All right. Uh, a very good afternoon from my side to each and every one of you who have joined the live sessions for molecular dynamics simulation. On behalf of BioInsight Solutions, I cordially welcome all of you to this webinar series, which will happen in a continuous fashion over next few weekends with different topics. So in today's topic, we are going to learn about the basics of molecular dynamics simulation, especially with regards to the protein and ligand simulations. Often during the drug discovery process, the protein ligand simulations are what is very important. And when it comes to the small molecular organic compounds which act as a ligand, getting their parameters right is a very important key process in this MDS. So let's learn some basics about the MDS and see how to set up the environment and run the molecular dynamics by using Gromacs software. Uh, Let me just tell you about us a little bit. We are actually a Mumbai, India-based bioinformatics service and training company, providing various kinds of bioinformatics services, especially in the domain of pharmacogenomics, computer-aided drug discovery, and related services, as well as we provide custom trainings to many institutes. Not spending much time on this, let's go ahead. Now, today's agenda is to learn about the molecular dynamics simulation. But as it is a very lengthy process, what I'll be doing is that I will not be focusing more main on the theoretical aspects or the biophysical aspects of this entire journey, but rather our focus will be on using the Gromacs as a tool to actually perform the MDS. So the initial part is going to be introductory and extremely brief. I'll just tell some of the most important keywords to be used during this MD. We'll continue with the applications of molecular dynamics simulation in various domains of life sciences, especially. And going ahead, let's talk about some of the important terminologies associated with that. One of the key aspects of MDS is using correct force field. So we'll have a very brief discussion on those as well. Again, with the practical aspect of it. Following that, we will be talking about the general steps which are involved, the flow chart which is used for performing the MD simulations. The Gromacs is a key software here that we'll be using. So we'll be discussing the architecture of the Gromacs, the different file formats which are used during the MDS with Gromacs, and how to set up the Gromacs simulation. Ultimately, after running a small simulation, we will also see how we can analyze this data by using VMD or some of the key commands within the Gromacs. So without wasting any more time, let's quickly start learning about the molecular dynamics simulation for protein ligand system. At this point, I assume that all of you have at least a brief idea of what exactly MDS does. When it comes to the protein molecular dynamics simulations or the DNA, it's pretty standard because most of these parameters and the preparatory steps are involved within the software that we're going to use. But when it comes to the ligand, we have to treat it separately, and there are many different steps we have to manually perform for the ligand. So we will be talking about these aspects as well. Okay, we already have a question. How to analyze the MMPBSA binding free energy? Ranak, that's a really good question. But let's see how we go about this because we don't have that much of time. It's an introductory session, so we may not be able to come in a full terms with the free energy calculations. But definitely, I'm planning another session with respect to probably next weekend on the free energy calculations, which are also important. Today is just about how to set up and run the basic simulation and run the analysis. So definitely, let's see if time permits, we'll definitely discuss those as well. So before starting simulations, one of the questions that often get asked, how reliable and how important these simulations are. Well, I think in today's world, everybody knows the importance and pretty much standard step is to run MD after some basic analysis. But the scenario has changed a lot in last couple of years, especially thanks to these three gentlemen, Martin Karplus, Michael Levitt, and Area Warshall, who were awarded the Nobel Prize in the chemistry for the very first time for the purpose of computational simulations or computer models. The Nobel Prize was awarded for the development of multi-scale models for complex chemical systems. So after this particular discovery and the Nobel Prize, the scenario has changed and the molecular dynamics simulations are becoming a very key aspect of all the studies, especially in the biological, biochemical, and the chemical domain. So we will talk talk about some of these things. All right. Again, we have some questions about protein-ligand dynamic simulations. Yes, we'll be talking about the step-by-step method, how to set up, how to prepare input, how to get the parameters, everything. How to analyze PCA. Yes, that will also be a part of the analysis part, how to generate the principal component analysis. Definitely we'll talk about it. Okay, now I'll be taking questions periodically. I may not be able to answer them right away because we have to finish some part before we get to that. So in case you feel that the questions are relevant and need to be answered right away, you can definitely push them into the chat box. But other than that, if you have some other questions, you can kindly keep it for the end of the session where we'll have a Q&A definitely. So to understand the principle of simulation, this is a very simple quote by Nobel laureate in physics, Dr. Richard Feynman. This quote kind of summarizes the what exactly molecular dynamics is all about. If we were to name the most powerful assumption of all, which leads one on in an attempt to understand life, is that everything is made up of atoms. I think we are already gone past that phase of just doubting whether everything is made up of atoms or not. We know that for the fact. And the next part of this quote says that everything that living things do can be understood in terms of the jigglings and wigglings of the atoms. So these terms, jigglings and wigglings of atoms that he has used, they refer to the continuous vibrations and the movements of the atoms that we see in the real chemical and biochemical systems. So the molecular dynamic simulation is rooted in the very fact that if we can understand these jigglings and wigglings or movements and vibrations, we can understand how they are going to behave. We can predict their next movements, and that's how we can understand the overall chemical reactions which go ahead with this simulation. Now, molecular dynamic simulation essentially is predicting how the dynamic behavior or the motion related behavior of the atoms would take place and predicting that by using some computational models. Essentially, the MDS methodologies are trying to mimic the behavior of different atoms, especially the multi-atom systems, where every atom is considered as a particle, an individual hardcore particle, and how they will behave in a certain environment. That's what we try to find. Of course, it comes with an assumption of a given potential energy function. So the movements of these atoms are dependent on the forces that they generate on each other. These forces in turn depend on the various kinds of interatomic and interaction potentials that these atoms have. So if you can correctly model these interaction potentials, your movements, the forces generated can be correctly estimated. So what exactly happens during the molecular dynamic simulation is that an energy function is used, which will tell us about various kinds of forces which are used by the atoms on other atoms. So if we know the positions of every atom in the given space and some basic properties of the atom, we can estimate different forces that they exert on each other. And by using the Newton's laws, which are there for the classical mechanics, but we can approximate them for the molecular systems. So we can essentially perform the calculation of the forces by using Newton's second law. And as you know, force is a vector quantity. It gives us the magnitude as well as direction. So with those directions and the velocities initially assigned to the atoms, we can estimate the movements of the atoms in a given time frame. That's what exactly the molecular dynamics is trying to do. The important part here is integrating these different forces and different interaction potentials together for the entire system. And that's where our MD integrator or algorithms come into picture. So with this, let's try to discuss some of the key aims of the molecular dynamics studies. Why do we perform the simulations? Now, simulations are very important because they escape or kind of avoid using actual resources and the real life experimentation to perform some of the property calculation, which can easily be done. So simulation can be inspired through a lot of things. There may be some experiments which are extremely costly or tedious to perform. Sometimes experiments can absolutely not be possible. Sometimes they are very dangerous experiments. So in all of these cases, you can go for simulations and perform the similar experimentation in the computational world. Understanding of these properties of whatever atomic systems you're using, especially in terms of their three-dimensional structure, is what is key over here. So it can serve as a complement for conventional experiments. You can replace certain experiments. It can, in fact, evoke some new experimentation from the data that we get. And even it can sometimes explain the experiments which are on their own not explanatory. So it tries to bridge the gap between the microscopic length and time at which various biochemical and chemical reactions happen and the macroscopic world of laboratory on which we observe things. So any observable phenomenon, let it be thermodynamic properties, let it be binding free energies, or any other like viscosity or diffusion and any other properties. So these are dependent on a certain collection of atoms at the microscopic length and their movements over the time. So if we can provide a necessary trajectory for this time dependent behavior, we can estimate those microscopic properties as well. That's the key here. So these simulations essentially provide guess at the different types of interactions the system is having and exact predictions of bulk properties. Now, the reason why I have used exact and the predictions at the same time is just that simulations have a capability to get to the exact calculations of the bulk properties. But unfortunately, because of the computational power limitations, we cannot simulate the systems long enough to reach to that equilibrium state. But let's see how that can be circumvented by using some statistical mechanical principles. These simulations are going to reveal many hidden details within the bulk properties that we can compute. So let's talk about some applications of this and how we can use it. So whenever you have any chemical system undergoing a chemical reaction, that can be because of various forces which are acting. That can be because of the chemical forces or nuclear forces. That can be because of the interatomic non-bonded forces. And all of this can be estimated if you can correctly model those interactions. So these kind of simulations will help us bridge the gap between the theory, different types of models which are currently available, and the experimentation at which we have non-ideal situations in most common cases. So during simulations, we can carry out many different types of experiments which are otherwise not possible. For example, working with extreme temperatures and pressures is a very difficult. Extreme temperatures are dangerous and the high pressure equipments are very costly. So if we already know the laws of this, thermodynamics and viscous gaseous laws, if you remember, so those already are there. So why not to use those laws to model the systems on a computer and predict the effect of various temperatures and pressures during the simulation. So those kind of experiments can also be performed during the molecular dynamics simulation. Now, when it comes to the actual solving these equations and what kind of interactions are taken into consideration, essentially it's a step-by-step solution to the classical equation. Now, the normal simulation that we'll be learning about are called as classical simulations because we use the molecular mechanics principles which are derived from the classical mechanics. So Newton's laws of motion or other laws which are especially defined for the physical bodies or physical objects, we are trying to approximate them for the molecular systems. But I think everybody these days knows that the molecular systems are governed by completely different set of rules, which we refer to as a quantum mechanics. So this is just an approximation, but still it can provide a pretty accurate estimate of many important properties when it comes to the biological systems. During this force calculation, as we know, many different types of interactions can be calculated. We have van der Waals interactions, which are derived through Lennard-Jones potential here. So here you can see the attractive and the repulsive term during the calculation of the forces on each other. We have Coulombic forces, which are there because of the different charges on the atoms. So all of these are combined into the calculation of these different types of systems. Apart from this, the energy contribution is also coming from the different intramolecular interactions, especially the bonded interactions. These are non-bonded interactions and these are the bonded interaction, in which the terms which are used during the force field involve the things like bond stretching and energy contribution. Because of that, the angle bending, the torsional rotations. So all of these together will be used in order to calculate the final force by using the force equations given in a force field. So here, as you can see, if you look at the Lennard-Jones potential, it essentially again we have attractive and repulsive forces here, but it essentially is kind of long range, so it can even act on the long range interactions. On the other hand, if you look at the Coulombic forces, they are very dominant when two atoms are very close to each other, but as the distance between the atom increases, they become very non-dominant and negligible. So of course, we're going to use some cutoff so that we don't have to go and for a lot of calculations, we can cut the calculations at some point. Now, the molecular dynamics algorithm, which whatever software you're using, it has different algorithms. But essentially, what it will be doing is that it will break down your time scale at which you want to perform the MD in a very discrete time steps or small time steps, usually at the level of femtosecond scale. So at every few femtosecond, you can compute the behavior of atoms and the changes which are happening there. So at every time step, what will happen? The forces on each of the atom will be computed by using these forces, which are calculated from the force fields. Based on the applied force and the direction and the velocities, the atoms will be moving a little bit. Every time there is any movement of the atoms, the entire set of calculations, the energies and everything will be recalculated. The new forces will be applied. Again, that will direct us towards the next movements of these atoms. And then this is the way the trajectory will go on till the time limit that we have set up. So this is what basically they do. So here it's a very small simulation to show how exactly they work and how we can use this in practice. So here is a very small simulation of a protein called valine, and the thermodynamic process which is being simulated is called as protein folding. Now, protein folding is a very key step when it comes to the protein structure, the 3D structure formation, and so on. So here there is a very small protein called valine that has been used. This is the actual crystal structure of the valine, the fragment of the valine. Now, during the MDS, only elongated amino acid chain was given, and the actual structure was not, of course, provided. And the amino acid chain was simulated for enough time, around 6 microsecond as per the simulation time. And at the end of the 6 microsecond simulation, you can see that it has actually folded exactly in a certain way, the way it would fold into the cellular system. And it will reach out to the same three-dimensional conformation, the way we can see in the crystal structure. So essentially here, not just the protein is being used, but along with the protein, the entire system is simulated, the aqueous environment with water molecules around, the ions to stabilize and neutralize the system, and everything. So this way you can simulate any chemical system as long as you have enough information about it, about the molecules used here. Now let's start talking about some key aspects of the simulation, some properties, and the terminologies which will be associated while we use the software like Gromacs. So in molecular dynamics simulation, the atoms are going to be in constant motion. And even if you see in the real life, all the molecules are always in a constant state of motion. That's how they interact with each other. That's how they form chemical bonds and reactions and perform the daily functions of our body. So even in MDS, these are under the constant state of motion. So they may not actually stay at the global minima, energy minima always. They may go from that and again go to increasing energies depending on how the forces are generated. The energy conservation is definitely followed in the MDS as well. As we know, the law of conservation of energy is universal. So here also the conservation is applied through the exchange of potential and kinetic energy. So at lower potential energy, atoms would move faster, having the kinetic energy larger, and vice versa. So the total energy of the system will be kept as constant. Now, during the simulations, unlike the molecular docking experiments, the water or solvent especially is a very important thing. Without solvent, we will not be able to get the thermodynamic properties as we would get in normal chemical systems. So these solvents can be given in the MDS through two different means. One is called as explicit solvent models and another is implicit. We'll take example of explicit as a practical example later. So explicit means you will be actually putting along with your structures of the proteins and ligands that you're using, you will also be using the structures of water molecules around to simulate the real system. The implicit solvent models will not really put the molecules there, but rather there are some mathematical models, the continuum model and others, which will use some statistical distribution to estimate the effect of water models on the simulation, and those will be used in this case. So both of them have their advantages and disadvantages. Explicit model will be much realistic, but implicit will be much faster because the number of energies and interactions we need to calculate will be much lesser than what is in the explicit. So in explicit, we have to use all of the water molecules. Coming to the applications of molecular dynamics simulation, especially with regard to the drug discovery, there are plenty of applications. The very first is determining how small molecules bind to their targets and how do they exert their effects. So during the protein and ligand molecular dynamics, you can understand how does a small molecule bind, what kind of changes are happening in the conformation of the protein, and how does those effects are being exerted. For example, here you can see here a system of a protein, a receptor, and a drug, which was presented in a Nature 2013 paper, where the allosteric mechanism of the protein function was elucidated by using the molecular dynamics. So in this case, the drug actually binds at this location, but the active site which is present at this gets affected. So to see exactly what kind of conformational changes are carried throughout the system, the molecular dynamics was helpful in elucidating the entire pathway of this reaction. Similarly, the molecular dynamics can help us understand the mechanisms of proteins and their actions, how do they behave, understanding the mutational modeling and what will be the mutation effect on the protein structure and the function, understanding the process of protein folding, which is very important. So by using this protein folding simulations, we can get the three-dimensional structures of the proteins for which we don't have the X-ray structures available. Apart from this, you can also use these experiments to understand the behavior of many molecules under various conditions, under temperature conditions, under different protonation states, and many other states can be studied during using this. Now, when it comes to the MD, we have talked about that most of the time we'll be talking about classical MD because we are using the classical equations to estimate those forces and energies. But we also have some other chemical reactions which cannot be estimated by classical MD because these reactions happen at the electron level scale. In the classical mechanics, we only have the bonds which are connected by atoms which are connected by the bonds, and that's all. We do not have any more detailed representation of the system. But in the quantum mechanics, the system is decomposed into the subatomic particles, and the electron level interactions are also found out. So these are just different types of reactions which we can study with the help of MDS and with different types of MD. So we can understand the polypeptide folding, biomolecular association between two different biomolecules or protein ligand interactions as we are going to study. We can understand the partitioning between solvents. We can understand the process of membrane or micelle formation in the lipid environment. Then we can understand many chemical reactions, enzyme catalysis, then photochemical reactions, electron transfers, and plenty of other reactions can be studied with the help of molecular dynamics. Now, for this first four type of reaction, these all kinds of reactions are governed by thermodynamic equilibrium. It is governed by very weak non-bonded forces of attraction between different systems. On the other hand, the next three reactions, these are often governed by the chemical transformations, and these are driven by the strong nuclear forces which take effect. Now, this first four type of reaction, they can be understood with the help of simple atomic degrees of freedom and the equations of motion. And we can estimate those by using classical molecular dynamics, the most standard one. On the other hand, for these reactions, we need to understand not just the degrees of freedom of atoms, but also the electrons and nucleus. How do they behave? How do they contribute, and so on. And that thus we need the quantum dynamical parameters, especially the Schrodinger's equation, to be used during this. So that can be used by quantum molecular dynamics. We'll see the limitations of both and the applications of both in the end of this session. Now, when we talk about the molecular dynamics, there are going to be many different types of terminologies which will be introduced. So let's try to get some of the most commonly used terms out of the way before we start the practical aspects of this. So first of all, during the molecular dynamics, we need to represent the system as realistically as possible, and to retain the properties of the molecular systems, we apply some constraints. These constraints that are applied automatically by the system, they make sure that the bond lengths, the optimized bond lengths which are used during the MD and the initial system, they are retained and they are not allowed to vibrate. If we allow these to vibrate, they will again have completely deflect the potential energy function because of these vibrations. So those constraints are applied. Now, why it is important to understand that these constraints come from the force field parameters. And if your force field that you're using is not accurate enough, then the lengths, and because of that, whatever forces will be generated, they will not be accurate. So that's where you have to understand how do the constraints are applied on the different systems. The next is the periodic boundary conditions. The periodic boundary conditions are often applied in order to minimize the surface effects. Everything that happens during the molecular dynamics is the play of different forces acting. So let's say we have designed a simulation box. Okay, we have designed a simulation box here, and in this we have kept our protein molecule, our ligand molecule, and all the water molecules around. Now, what will happen whenever these atoms are moving? They can move in any direction as per the forces applied. So, if they move towards the wall of this box, this is the limitation, the boundary that we have set for the reaction to happen. Now, this will introduce some surface effects because of the collision. Whatever velocities they have, depending on that, the collision forces will be introduced, and that will interfere with your calculations of your actual system forces. So, to reduce the surface effects, we apply the boundary conditions. Especially in boundary conditions, we create a copy of our main primary simulation box in all the three dimensions, not just here, but in every XYZ directions, you can generate the copies or images of the system. These images especially help us reduce the surface effects, and also they can help us in bulk property calculation. So essentially what happens in any case, if the atom is going towards the wall of the container or this box, they will not be colliding. They will simply move to the next image of the same box. So when this atom moves to this, exactly same atom from every box is going to move out. So this atom from this box will move out. It will come from this side and enter onto this box. This will also move, and accordingly, it will be a continuous flow of atoms without any surface effects or any collisions of the wall. So for that matter, periodic boundary conditions are very important to be applied onto the system. Second, we'll be talking about a neighbor list. Now, neighbor lists are the list of atoms which are very close to each other during the system simulation. So if these two atoms are staying very close to each other for almost all parts of the simulation, then we don't have to recalculate their forces every now and then. On each other, whatever forces they have, they will be kind of constant as long as they have the same distance. So by using this neighbor list, there are multiple methods to use the neighbor list. We can reduce the number of pairwise calculations, which will be much more economic and faster for algorithm to run. Second thing is the time dependence. So it's important that you allow the system to run for as much possible time as you can. Again, we always have these limitations depending on how much computational power you have, but that can be also set. So this time scale that we set may depend on what kind of phenomenon that you're trying to find. So if you're simply looking for the refinement of a dock post, you can maybe do it for maybe few nanoseconds and that will be done. If you're looking for the very large movements like loop movements in a protein or domain movements in a protein, these kind of movements happen over the period of maybe microseconds or milliseconds. So you have to allow the system to simulate for that much amount of time. If you're talking about a very quick chemical reaction, those happen at the like very quickly on the scale of nanoseconds. So you can perform the simulations as nanosecond. So depending on the behavior or the properties you're trying to estimate, you are expected to set up your time scale, and always it can be extended as per the requirement. So these are some key properties of the molecular dynamics simulation. When it comes to the limitations, your limitations can come from many sources. First of all, time steps. So we will talk about these time steps again when we see the input parameters. So time steps are basically at every how much time frame the calculations are going to be performed by the uh, dynamics engine. So usually these time steps are required at a very, very minute scale to retain the numerical stability of integration algorithms. If you don't use it such a small time scale like at the femtosecond level, the numerical stability will be lost and the integration that it's being done will not be proper. So usually we use like 2 femtosecond or 1 femtosecond. Another reason for these time steps to be considered is also the vibrations of the atom. If you look at the smallest time scale at which some atomic movements happen, they usually happen at the femtosecond time scale. Like hydrogen bonds, which are very light and small, they vibrate much with a higher frequency. So they can happen these movements at the level of femtoseconds. So we have to keep it so that we can also capture those movements during the simulation. As we discussed about time scale, these can depend on multiple parameters, but Also, you have to remember that more the time scale that you increase in time, the actual simulation time. I'm not talking about the computational time here, but then these time scales will also increase your simulation calculations. So when you're looking for nanosecond scale simulation, you are already requiring millions of calculations to be done. When you go for microsecond or millisecond, you're talking about trillions and zillions of calculations, which will require to have a very powerful computing machines in order to perform the simulation. So that is the limitation that you will have to put with respect to hardware and the time scale that you can have. Second, the next thing is the force field accuracy. As the different forces and everything that is coming up is dependent on the correct calculation of the force field parameters. So your force field that you're choosing has to be accurate. And anyways, force fields themselves are the knowledge-based data sets, and they are coming from some approximation. So you have to make sure that you, from the literature, you are researching correctly what kind of force fields are used for certain systems, which is good for protein, which is good for DNA, and many other stuff. And second thing, during the classical simulations, we cannot estimate the formation or breaking of bonds. That cannot happen because covalent bonds are dependent on the electron level interactions, which are not considered in classical MD. For that, we of course have to go for the quantum mechanical MD for this set. For performing the molecular dynamics, there are many different types of tools available. We have Amber, NAMD, Gromacs, Desmond, SMD, OpenAM, Charm, LAMPS, and many tools are available there. Out of this, some of the most popular ones are Amber, NAMD, and Gromacs. Desmond is also nowadays popular because it's also freely available now. So these are many softwares which run in a different way. Some of them are costly. The Amber, SMD, these are the paid and commercial softwares, while other in the green, these are the open source software for the scientific and research community. Now, the core functionality of all of these softwares is, of course, the same. It just depends like what kind of force field are they optimized for, how do they differ in their speed, the scalability, especially with respect to different site systems, the cost which may be involved in some of them, and feasibility of using them with the current hardware. We also have some softwares available for the visualization and analysis like VMD that we'll be learning today. So today we'll learn about the Gromacs and VMD, how to set this simulations and how to run them by step-by-step manner. Now, as I mentioned, there are two types of simulations we can do, the quantum and molecular mechanics or classical simulations. So quantum mechanical simulations, they require to solve the Schrodinger's equation for the given system, which is often a very difficult process. We cannot do it for a very large systems, but for very smaller systems with few tens of atoms, we can definitely perform. They definitely give you a much accurate results of the simulation, but they can currently be applied to very small systems, maybe up to 100 or few more atoms. And this system can be simulated for around 100 to 200 or so picosecond. I'm talking about the most decent workstations, not supercomputers. Molecular mechanics, on the other hand, they are based on the empirical forces calculated by the Newton's laws of motion, which are approximated for the molecular systems. These are less accurate, especially when it comes to the electron level interactions, but they are practically very fast and we can perform even for a very big system. They have been done with the millions and millions of atoms. The simulations have been done so far. So they can be used for biological systems, which are often much larger in size as compared to the small chemical systems. We also have the mixed quantum and classical simulations, where some part of the system we can treat with the classical simulations and some part we can treat with the quantum simulations. Especially when it comes to the protein-ligand simulations, ligands can be treated with quantum parameters, and the protein can be treated with the classical parameters. Or even the protein's binding site can also be added to the quantum parameterization, but it is a little bit tricky to perform so. So those kind of hybrid QMMM simulations are also done. Before going that, let me just see some of the questions have been posted. How to verify the aptamer structures that modeled computationally is valid? Okay. Let's come to those questions later, which are not directly related. How to calculate the average values, RMS, RMSD, SSA? Yeah, we'll be talking about that during the analysis phase, Zeshan. We will come to that. How to correct the poor rotamers and bond length in protein modeling? Well, we can subject it to the rigorous energy minimization, especially with solvent minimization. Again, we'll take an example of that. That's a good question. So during the preparation of the system, we'll discuss all these characteristics. All right. So when it comes to the requirement of using the molecular dynamics simulations on your computers or on your institute, it's important to understand that these are extremely computationally expensive studies, and you cannot perform those on your daily desktops or laptops. Maybe small simulations you can, but you need a very powerful dedicated machine to use those, or you need a specific hardware like graphical processing units or GPUs to perform so. There are many different facilities, academic as well as commercial, are available for you to use this kind of processing power, which can be taken. Or if you already have some facilities, you can always reach out to them. So this is for the Indian participants. We have the government of India who have a dedicated bioinformatics, the supercomputing facilities called BRAF, and then we have the other SCF Bio and others which can be used freely for the academic community within India. So you can get a very powerful nodes to process your simulations, or you can also approach companies like NVIDIA for GPU grant to get a powerful machine for your own usage if you have a good proposal. There are also some like commercial facilities or toolkits available like SMD or HTMD for high throughput molecular dynamics and so on. So which can also be utilized by people if they do not have the enough resources. Okay. What are the softwares used for the MM and QM methods? Well, all the software that we have mentioned, all of them can support both MM as well as QMMM and MM simulations. So there are some things where you can plug in. For example, quantum mechanics, we have many other software like ORCA or MOPAC, which can be integrated with Gromacs or other softwares, and those can be then put into together for hybrid simulation. So those can be used. Again, we have things like Gaussian, which is for quantum mechanical simulations, and molecular mechanics. We have all of the things which were listed. So now let's talk about how to set up a molecular dynamics simulation. What are the steps involved? What considerations we have to go through? And after that, we will see practical aspect, how to use the Gromacs other toolkit to do all those things. These are some of the common steps or common protocols involved. It may not be exclusive depending on your case and the system. They may be deviating a little bit. Some extra steps may be required, or some of the steps may not be required. So let's go through some of the steps involved in the simulation of protein and ligand system. The very first thing you need an input system or the system building. Now, system here refers to whatever chemical reaction you want to simulate. You have to create the most appropriate representation of that as you can. So, for example, most of the times this system will come through the X-ray data or X-ray structures that you have either from PDB. They can also come from modeling or theoretical models. For example, the protein-ligand interaction reaction can come from the molecular docking. So, whatever output you get through modeling the interactions from molecular docking experiments, those systems can be used as the initial system. Or if you want to do the protein unfolding simulation under certain conditions, you can use the structure of the protein either modeled, you can have that. If you want to predict the 3D structure of the protein folding process, you can randomly create the amino acid polypeptide chain by using any building software, and that can be used as your system. So if you are using a membrane protein simulation, then you have to generate a membrane and a protein embedded into that. So that can be your initial system. So we have to report this initial system as a set of coordinates of atoms in a proper manner to the simulation software. If you want to do the photochemical simulations, those kind of atoms can be your system. If you are doing nanoparticle simulations, those can be your systems. So depending on what reaction you want to follow, you can create your own systems by using multiple different types of methods. Once we identify the system at hand, mostly for the protein ligand simulation, this comes from either the bound complexes from the PDB, or more commonly they come from the molecular docking experiments. So after performing the molecular docking, the complex which is formed, we submit it for the MDS, and that will be the system that we are talking about. The system preparation goes through multiple different steps. So we need to make sure that the protein which we are using and the ligand which we're using are there in the proper state. All the atoms, the hydrogens especially, are properly formed and attached. We can use multiple preparation softwares for that. We need to generate the topology files, which will explain from the force field parameters that what is the topology, what are the bond angles, bond lengths, the torsional angles for every set of atoms currently present in our molecule. For proteins, these topology files can be easily generated because these data is already embedded within the force fields. Then we need to solvate the system because we are going to run the simulations in some kind of a solution. So we need to solvate it. Now, most commonly, the solvent is going to be water because most of the reaction at the biological scale happen within the aqueous environment. Or you can also use any other solvent like methanol or paraffin or anything that you want to carry out for your reaction. The charge collection can also be done, especially we add different salts, the way we add them in different buffers to make sure that the overall system is neutralized. It is a requirement for the protocol which calculates the long-range electrostatic interactions called PME or particle mesh evolved, which will need the neutralized systems to be performed. If you're using a ligand in the sense any non-standard residue you are using, like for example, the proteins, DNA, some common ions, some common metal ions for which the topologies are already present in the different force fields. But when ligands come into picture, these are novel molecules. These are different. There are no set ways of defining them. So when it comes to the ligand, we have to treat it separately. We have to get its topology generated either from some software tools or we have to manually generate that topology, which will convert to the rules of the force field that we are using. We'll see some of the options for this as well. Once we prepare this initial system, so initial system will consist of your solute, your solvent, your ions, and ligands if they are part of the system, and that will be ready for next stage. If we directly take this system for a simulation, it will create a lot of artifacts. There will be many problems because currently the system is randomly generated and it is far away from the equilibrium. So we need the system to be as close to the equilibrium as possible before we run the molecular dynamics. So we have to make sure that we run it through various preparatory steps, which include the energy minimization, in which we make sure that all the atoms that we have added, the solvent, the salts or the ions and the proteins, their side chains and all of them are oriented properly with respect to each other. Steric properties are checked over here and steric hindrances are resolved during the energy minimization. Once the system is minimized, it can be taken for the next stage of heating and equilibration, during which we make sure that all the atoms are at the correct level of heating, whatever temperature at which we want to carry out the reaction. And equilibration will make sure that this proper equilibration is reached. So it will make sure that the pressure, the temperature, and the volume, the density, everything is matched with the real system. So equilibration will take care of that. Often if we don't do the equilibration and directly run the MD, there are formation of vacuum bubbles and some unwanted forces which may fail or crash the simulation because of the unrealistic forces generated. So these are the different stages through which we have to go through for setting up the MD. When it comes to the actual preparation, we have to generate a lot of different files, a lot of different parameters, and consider many properties of the system before we set up the simulation. So input generation is very important step where you get your three-dimensional structures of your proteins of your systems by using different methodologies. Then the topology can be generated for proteins within the software that you're using, or proteins or DNA or any other common atoms. But for ligands, we have to generate the topology separately. The energy terms will come from the force field, so we have to select the proper force field for the process. Now, the next will be selection of the interaction model. How do you want to define the interactions? Which type of models you want to use? Within given software that you're using, there can be many options for you to use. So you can choose which model to use. How do you want to apply the boundary conditions? Whether you don't want to keep them or you want to keep them just in the X dimension or XY or XYZ, all the dimensions. So when you have a cubic box and the XYZ periodic boundaries, essentially you have a 26 copies of the same system all around it, total making 27 images. If it is cubic, but cubic is just one type of a box we can have for simulation. We can have octahedron, we can have a sphere or shell, and many other shapes can be used to create the simulation box. Then at every step, we have to identify what kind of thermodynamic ensembles we have to use, whether you want canonical or non-canonical. Basically, they refer to the isobaric and isoporic and other type of processes in the thermodynamics. So the commonly used ensembles in the MDS are NVE, NVT, and NPT. Each one of this corresponds to the N is for number of particles. So in each of the simulation, as you can see, the number of particles is always going to be constant. Our system is not going to change here. V in NVE, the volume of the system will be constant, and E, the energy of the system will be constant. In NVT, you will have the volume, temperature, and particles will be constant, and NPT, the number of particles, pressure of the system, and the temperature will be constant. So you can use these different ensembles depending on different properties you want to use. We'll discuss this during the practical setup. Then you have to select the target temperature. At what temperature do you want to estimate this characteristic? So you can set that. So once you, then what pressure you want to give this. So pressure can be applied during the parameterization. Then selection of the integrator, which MD integrator will be good, which type of integrator will be good for this system. You can explore this by reading the documentation of the software that you're going to use. Then which thermostat or barostat can be used, what are the available options. Again, these are everything of these is given properly in the documentation, but we'll talk about some commonly used systems within this. Finally, you will perform the production run after the system is properly equilibrated. The final main MD run is called as the production run, in which you will be collecting the averages, the thermodynamic averages over the time that you have set in the MD simulation. And these collections will lead to a proper analysis and calculation of many different properties for the given system. Okay, so these are the very generic steps, and we are going to go through all of these steps for a given system. Okay. So now let's look at the Gromacs as a software. What properties does it have? How does it use different file formats to represent all these different things that we have talked about, and so on. So let's go ahead. Okay. Now, okay, there is a question. Can you get us Gromacs and its installation process? This will help us practice. Definitely. So what I can do right now, I'm not going through it, but at the end of the session, I'll explain how you can install the Gromacs, and I will also share a document with you which have a proper installation procedures, the steps both on Linux as well as on the Windows. Gromacs, I'm going to introduce what exactly the Gromacs is and how it is used. Gromacs essentially is a very popular, very old simulation package, and it has been there for a really long time. And it has evolved itself into a really good and popularly used software package. It's a Linux-based system, so you have to install it within the Linux system. It's a command line-based software, so you have to access it through the commands only. And because of the recent advancements into the Windows 10 operating system, even Windows can now support the Linux subsystem, and that can be used for installation of the Gromacs. So I will give you the step-by-step guidelines to install this Gromacs both on Linux as well as on Windows. So you can make use of this. So on the website of Gromacs, you can either use the direct source code to compile on your own if you're familiar with the command line applications, or you can directly go for the packages which are present in your package manager. Now, Gromacs is very fast, very accurate, and it can also support the GPU acceleration, which is graphics processing unit, which will make your simulations much faster. So this is very important in many simulation packages. Having the good graphics card will definitely help enhance the simulation. The documentation of the Gromacs is really good. You can go through almost every parameter which is available, its importance, and how it can be used in different ways. So all of this can be available here. But anyways, we're going to see how to use it. So the next part we'll be learning about is the Gromacs software and the file system. How does Gromacs processes the data? What kind of things are available, and how we make best of use of that. So Gromacs, as you can see here, is a software which is usually on the scale 3 to 10 times faster than other MDS packages, depending on the scale of the system. Apart from Gromacs, NMD is another good software that can be used to associate, scalability with the size of the system and the size of the hardware. You can easily scale those. Very user friendly. Again, if you're not familiar with the command lines, it's not going to be very user friendly. But even on command lines, it provides a very detailed analysis of the errors or the warnings that are present in your system, and it can guide you how to change or what exactly to modify depending on your requirements. It allows you to perform the formation of the trajectory in many different formats, and not just that, it also provides a very basic analytical tools within the software itself. So when you will install Gromacs, it will be installed as a software suite with huge number of commands, huge number of programs for preparation, for setting up, for running, for the analysis, and many other things. So during all of these things, the file formats are key, and it uses many different files for many different purposes. And this can be confusing initially, but once you run your initial MD, you can always run it once or twice and test those small systems, and then you can scale your systems for the larger size. So some of the common file formats which we will be associating with Gromacs are as follows. The first is the PDB file, or the protein data bank file. This, I think you will be familiar with. It's a basic coordinate format required or used by the protein data bank to store the coordinates of the atoms in a given system. So it is used for the input. So your input will mostly be coming from the PDB format. The next type of file formats which are used in the Gromacs are topology, or the extension is .top. These topology files contains all the force field parameters. So what exactly the preparatory steps do? They analyze what kind of atoms and residues are present in our system. They match those atom types and residue names to the names of the residues in the force field files, and accordingly they will write the topology of your system into these files. This is very important file, and we'll be dealing with it a lot. The next are the .gro files. So these are the basic coordinate files for the Gromacs. So internally, for all the transformations and calculations, Gromacs uses this format, which is converted from the PDB file, and that you can use. So the .gro file format contains same thing, the residue numbers, the names, the coordinates, just like PDB, but it also contains these positions and velocity information. Now, these positions are transformed into such a way that the calculations are done at the nanometer scale. So instead of angstrom, the default for PDB, here we have the nanometers. And the velocities are also reported into nanometer per picosecond scale, so that those can be reported. So again, these files can be generated directly from the software itself. The next type of files the many commands in the Gromacs require are the TPR files. These files contain the starting structure. They basically compile all the input things, your molecular dynamics parameters, your topology file, your growth structure, GRO structure. So all of these parameters, it compiles into a single binary file, which will be then used as an input for actual molecular dynamics run. We'll talk about these commands and which type of commands or programs are available through this. There is a question, can we run it on Windows? What should be the specification of a laptop to run the MD simulation through using Gromacs or Desmond? Yes, I'll come to that. Definitely just hold on to this question. I'll definitely discuss this. It's a very important question. So at the end of the session, I'll discuss how to choose the configuration and whether it can be used. Desmond cannot be used on Windows, but Gromacs can be used through the system. The Desmond is very difficult to use on Windows. Okay, so the next few file formats are the output file formats, which are generated by the Gromacs. So one of the file format which we'll often encounter is the TRR file format. This is nothing but the trajectory file. It basically reports every now and then the coordinates of the current state of the system, and that will form the entire trajectory of the simulation. The simulation also outputs the EDR files. These files are containing different types of energies of your molecular systems and groups. So those can be tracked. So for plotting, checking the energies, the graphs and all that, you can use the EDR files. These EDR files then further can be converted into the XVG format, which is often used by the programs like XMGRESS to plot the data on the graph. Then XTC is similar to TRR, it's just the compressed and the compact binary file format for the trajectory. So if your TRR file becomes too big to handle or manage, you can convert them into XTC, which are the compact format or compressed, and then you can export it and easily transport it. Now, another very important file formats are the MDP file. The MDP file is the key here. For every step in this process, you have to create a molecular dynamics parameter file. This parameter file sets up your experiment, and there are thousands and thousands of parameters available within the MD, and most of them are key parameters that you have to set. So these parameters look something like this. So for example, general energy minimization, this will look a huge list. These are nothing but, like what kind of things you want to use, which integrator you want to use, what should be the time step, how should be the total time scale at which you want to run the MD, how often do you want me to write the files, how often do you want me to write all these different parameters. We'll discuss those with the example files and all of this. Going ahead, it also has the parameters like what kind of interaction model you want to use, how do you want me to calculate the Coulombic and long range electrostatic interactions, how do you want me to handle the neighbor list, and a lot of parameters. What should be the reference temperature? How do you want me to couple the temperature and different groups in the system? So what kind of model do you want to use? For example, in this case, you have, so this is a very generic file that is available. So you can see here how exactly you are talking about different coupling parameters for pressure, for temperature, and all other things. You also have parameters like velocity generations, whether you want to generate or not, and many other things. So we will be talking about some standard parameters. The next part of the process is using a force fields. Force fields, as we have just discussed, these are the set of equations or potential energy functions which provide the necessary parameters for the given system. Now, the Gromacs by default supports many different force fields. It has the Amber series of force field. It has its own Gromacs series of force field and Gromacs force field. It also has a OPLS force field and few others. We can also integrate any other external force field which can be compatible with Gromacs, like Charm force field. You can use. You can download these force fields from their respective websites and use it directly during the MD. So this way you will be able to set up and choose. We'll talk about which are good for what system. So in general, like the Gromacs force field uses the united atom models, not all atom models. So they kind of combine some small atoms like hydrogens with their parent atoms and use it for faster simulation. So we'll use this just to make sure it is quick. The next, we have Amber force field, which are very good for the protein and DNA, RNA, or nucleic acids essentially. These are optimized for the biomolecules. We have OPLS model, which is really good for the proteins and hydrocarbons, and they are also all atom models which can be used for any liquid system. So they are very good for liquid systems. Most other force field assume your systems to be gaseous system and they try to apply those kind of parameters, but OPLS is good for the liquid systems. The Gromacs, when you install, it comes with a plethora of different programs within its suite, and these programs are as follows. Some of the important will be using PDP to GMX, which will help us convert the PDP file into the Gromacs file. So it will generate the topology and many other things. Then we have editconf for generating the simulation box of a certain size. Then we have a genbox command or solvate command to fill the box with water molecules. So we will be actually performing each one of these steps, understanding the input, understanding the command, and then visualizing what exactly is the output of the command is by using VMD at every step. The two key programs we are going to use often are the GrowMPP and the MDRun. So GrowMPP and MDRun, they offer kind of work in couple. The GrowMPP command is a preprocessor program. It will compile all the input files, the topology file, the MDP file, and the .gro file. All of these files combined, it will create this TPR input file, and this file is required by the MDRun program, which will actually run the molecular dynamics simulation or integration that you're actually performing. So this TPR file can be generated from GrowMPP, and this can then be given as an input to many other programs. The MDRun is the main program which will run the molecular dynamics or energy minimization, depending on whatever you have. So it can actually perform many things. It can perform the Brownian dynamics, Langevin dynamics, conjugate gradients, deepest descent minimization, and so on. It can read the TPR files, and depending on the files that whatever input we have given, it can create the different neighbor list from the file and use those to calculate the forces. Finally, these forces will be summed up globally for every time step, and those will be reported as an output file. The output of the MD run is three types of files. One is the trajectory file, which will contain the coordinates over the time. The structure file, the final structure of the Gromacs in the GRO format after the MD run, and the energy file, which contains energies, temperatures, pressures, which are documented over the time scale of simulation, whatever time we have used. Apart from this, it also creates a log file, which shows the log of the run, like how it has run, what are the different steps and everything. And many other programs are there, which we will come to just in a minute. So now let's start looking at the molecular dynamics simulation practically. To use the molecular dynamics packages in Gromacs, the installation I'll come to a little later, but once you have installed, they will be activated on your command line. So you have to have a little understanding of the command line to use this Gromacs. I'm not gonna have a separate class right now on the command line, but just to give you some basic idea, command line applications are the interfaces through which we can talk to many programs which can be accessed through commands directly, not through graphical interfaces or UIs, but directly you can interact with them through commands. These commands can take inputs, they can process outputs, and many other things can be done. Some basic commands using the terminal, like on Windows, we have the CMD, which is command prompt. On Mac, we have terminal. On Linux or Unix-based machines, we have this kind of a shell, bash shell or CSH shell, so on. So this is how it looks like, and this is how we are going to access our entire system. So there are a few common commands like ls to print the output of all the contents of the current folder. So currently I'm in my home folder, which is shown here. To change the folder or navigate to any other folder, you can use the cd command, which stands for the change directory, and you can basically reach to any other directory on your computer and see what kind of things you have. There are many other commands, basic commands like editing files by using vim, copying, moving files, and so on, that are also available. You can just search on YouTube for a very quick revision for the command line applications, and you will get all this data. So the system that we are going to use for the practice here is made up of a protein and ligand after a docking. So first I'll show you the output. How does it look like? Okay. So I have here a few files which I already have generated, especially the input and the key protein files, and the rest of the things will be generating by understanding each step during the simulation process and see. So very first, let's check out what exactly our initial system looks like. Our initial is simply a protein and a ligand which have been docked with each other. So I'm going to show you this through the VMD, which is a visual molecular dynamics software, which is used for visualization and analysis of molecular dynamic systems. So let's check what the initial system looks like before the simulation. I'll load these proteins. First, let me load the protein. Load it. Okay, so this is the protein that I have, and the docked ligand position is this. Okay. So that is the ligand. Okay, so ligand has been added. So let me just quickly show you the system before simulation, and every step we will check that. Now I'm just going to change some of the representation here of this. So let's show this as it is, and the protein I'm going to show as ribbon representation. So you can see. So this is a system we have currently, and we are going to simulate it. So the aim of this is to understand the binding dynamics of this molecule and the protein, how stable it is, what interactions does it have, which are stable over the range of simulation, and does it stay in the binding site or move away, what conformational changes happen to the protein. So these are the type of answers we are going to get from this simulation. So let's close this and start running the processes. How do we go about it? So for protein preparation, the stages are very easy, but for ligand simulation, it is going to take some extra efforts for us to go through this simulation. So these current files, as you can see, are in the PDB format, and as you know, the PDB format essentially contains the coordinates of every atom which are present in the system. and few other remarks which might be there. So it contains the residue names and the coordinates of every atom. So the first command that we are going to learn is the PDB to GMX command. This command is the first command that will prepare or generate the topology for the protein. Okay, now we have a protein and ligand in a two separate PDB files because we need to prepare them separately. Ultimately, of course, we are going to combine them together, but for now, they need to be separate. So let's learn the very first command of Gromacs. So how do we access the Gromacs program from the command line after installation? So it's very simple. Once the installation is successful, you can access the Gromacs with just a command called GMX. So GMX is basically the main Gromacs command. If you directly put the command without giving any parameters or any options, you will see that it throws some output that, okay, this is the correct way to use the Gromacs. It requires these different options. If you're not sure how to use any of these commands, you have many help options. So for example, you can take help and see what kind of commands are available just by typing the Gromacs help and commands. So it will show you all the commands which are available in the Gromacs package. So each one of these commands is a separate individual program within the Gromacs. So you want information about any specific program, let's say PDB to GMX. So you can again go for GMX help and the command name PDB to GMX. It will throw all the help that this is available, like how to use it, what exactly it does, and a lot of documentation is available. It's very good, so you don't have to worry about if you miss something or don't remember something. When if you are a beginner in this field, of course you don't have to remember everything as it is. Over the time, you will get practice of getting the patterns in these commands, and then you can use this for your calculations and so on. So let's run our first Gromacs command. Let's first check our data. So the folder contains some MDB files, which are the molecular dynamics files for every run we will see. Then there are two PDB files, which are the actual input structure files, which are there. And again, few more MDB files. So let's run the first command called PDB to GMX. This command takes input your PDB file. So it can be given by the parameter -f or -f, which will take the input file. So F here stands for the file. And this information, again, you can take from the help documentation. The important here is mdm.pdb. Now remember that our protein and ligands are named like this. The protein file is mdm.pdb. This is the protein called MDM that I'm using here. And K23 is the name of the ligand that I am using, which is bound to this particular protein. Now these are separate currently, but we're gonna, after we convert them into topology and the Gromacs file, we will come back and combine them. The next, it requires the output file. So you can give it through the -o command. So command line, as you can see, the pattern is followed for every command. It starts with the first of all GMX. That should be your first word. Within the GMX, which program you want to use, that you have to give, and then the inputs and output parameter for the program. So how do you want to save the output file? Let's call this output file that it is going to be generated called mdmpro.gro because it's just a protein currently. And this is the minimum things it is required by the PDB to GMX. When we will press enter, it will start running the program, and it is an interactive program. So it will ask you first, which force field do you want to use for the current system? As you can see, there are plenty of force fields given, and there are some basic description like amber, which is there for nucleic acids and proteins, amber optimized for nucleic acids. Then we have charm 27, but currently the charm 36 is a new version, so that can be used. You have the Gromacs force field, then you have OPLS force field, and so on. Just to show the demonstration and a very quick way of getting the results, I'm going to use the Gromacs force field. So it is a united atom force field, so we can get a quick results. So I'm going to use that. But for best results, you can always use OPLS. It is a very good force field for the protein and other organic molecule systems. So that can be used, the 15 number. So whatever force field you want to choose from this, you can simply press the number of that particular force field. I'm putting 9 here just so that I get the Gromacs force field, and then enter. Once you enter, it will show you the supported types of water models. So what kind of water models you want to include during your preparation. So that can also be done. Okay, now how do you know about all these things? How do we remember? Again, as I said, don't remember always. Simply go to the help, and when you go to the help, it will show you every parameter that you need to know. So as you can see here, these are the some common parameters. The very first input required is the F, that is the input file. O can be the name of the output file. If you already have the topology, that can be given. Then input, the topology can be given. The ITP force field file can be given. If you have the indexes, we'll talk about them because those can be given, and many other things. But apart from that, you have options like do you want to separate the chains? Do you want to merge multiple chains into the single chain? Which force field you want to use? You can also give it through command line. Which water model you want to use? That can also be given on a command line. Then so many other things are there. Whether do you want to keep it interactive or automated? You can have things like understanding the residues like lysine, arginine, aspartate, glutamate, glutamine, histidine. There are protonation states which are key in titrating the protein correctly. Those can interactively be set. Now, most common cases, it will be expected that you are using the pre-prepared proteins. Those should be titrated well with the proper hydrogens according to the whatever pH at which we want to use the simulations. So if you want to, mostly the titrations will be done at the physiological pH of 7.4. You can do those through series of software like Gromacs, or there are some online tools also there for preparing these proteins for simulation like Protein Prepare and so on. But in a very expert cases, you can manually select the protonation state for each one of these amino acid based on your understanding of their protonation states. You can also choose to add hydrogens by using the H option if they're already not there. You can also choose to ignore the hydrogens if they're not there. So this basic pre-processing you have to do with the hydrogens and any other things when you give your input system. Anyways, that's part of your earlier pre-processing, docking and everything. So let's not worry about that. Now let's run the command for our system, that is MDM PDB file that we just checked. It doesn't contain hydrogen because I'm using the Gromacs force field, which is anyways united atom force field. So I will select it. Then the water models would look something like SPC, which is a very simple point charge, most recommended for the united atom force fields. So I will select the one recommended, one number, and enter. So once I enter this command, it will give us some very important information about the system. Before cleaning and after cleaning, how many total things it has checked, the dihedral angles, the torsion is corrected, and everything. Most importantly, it will tell you the total charge on the system. This is a very important output. You have to remember it. It's okay even if you don't remember. This is anyways going to be written into the topology file. You can always find that again with that. So currently the system, the protein has a +5 charge, and we are going to need to neutralize it in the later stage before we set our platform for the simulation. Now it is writing the topology file, it is writing the coordinates file, and it will generate 3 files in total at the end of this run. So apart from this SPC model that we use, there are many other type of models like TIP3P, TIP4P, which use different types of protein models or water models for this particular purpose. Okay. Now, the next, once we get this file, we need to check everything that has been written by the software and make sure that we understand all of this. So now the new files which are generated are as follows. One is MDM pro.grow. This is nothing but the GRO file of the coordinates which confer to the atom names as per the force fields which are used here. So we can see all these coordinates and everything. The next, it has generated the topology file, which is a very important file, and this is what we're going to need in every iteration of these different commands. So the topology file contains understanding of your system, your atoms, your molecule types, and their respective values like charges, masses, and so on. So based on the force field that we have used for the protein, that is the Gromacs 43A1 force field that I have used here, it has used one molecule type, that is protein. each one of the residue and the respective values are given, the coordinates and the charge and the mass. The total charge on each residue is given. At the end of this entire list of all the residues which are given, you can see the total charge on the protein, which is +5. So here also you can see the charge. In case you forget to look at the command line earlier, or if you forget the charge, you can come back to the topology file and look at the charge here. It has the parameters from the force field, like bond lengths. The different bond lengths are there. After the bond lengths of every atom pair which are bonded, then you have the atom pairs apart from the non-bonded pairs. Then we have the bond angles. These are the different angles between different bonds. Then we have the torsional angle. So all of these parameters of the protein are readily available in the force field. So that's why the topology file has been easily generated. But when it comes to the ligand, we have to generate this topology either manually or by using some other software. So when you come to the end of the topology file, it will have very important information. It's a very big file, but you can directly come to the end of this. So here it will show you what other things are being used apart from the protein topology. We are using the water topology because we are using the water models. We are using the ions topology because we are going to add ions as well, but there is no ligand topology, which we'll be adding a little bit later. And the molecules directive, each one of this is called as a directive of the file. The molecules directive will enlist every molecule that you have currently in the system. At this moment, only thing we have is a protein chain A, that's all. Now, every time we'll be adding a new residue, new water, new ions, anything, those will be automatically enlisted one after the other at this. So every time topology file is the one which is going to get updated, and then you can check that file for everything that is going correct or not, for all the atoms are considered or not, to add the topology of the ligand, to add the constraints and all of these for ligands. So this file is now currently generated. I'm not going to change anything. The third file which is generated is called as posre.itp. Now this file is the positional restraints for the given protein. Now positional restraints are very important. During our initial preparatory steps like minimization, equilibration, heating, we are going to keep the restraints onto the protein. The protein is not going to freely move, but its atoms can only move in a very local limited dimension. So these are basically the different force constants given in each dimension around which they can make some changes. This is automatically generated for protein. Again, we have to generate the same file for the ligand separately manually. That's how we have to work with ligands, and that's why ligand simulations are a little bit complicated than simple protein simulation. So our first step is completed. We have successfully generated a PDB to GMX file. We have converted things into the Gromacs. If you check the files, we have the mdmpro.gro, which is converted. Now this is the file we are going to use for the next stage. Now the next stage is going to be generating the simulation box. Currently we have not changed anything, just the format has been changed. So the next stage will be to generate the simulation box. For that, the command we are going to use is the edit-conf command. But before that, let me just prepare the ligands as well and show you how to include the ligand topology and everything. Now let's come to the ligand. Our ligand contains a non-standard residue. If you look at this structure, the structure contains only a single ligand with some coordinates, and the name of the ligand is K23. So this is the name we will be using to refer to this ligand always. Now the ligands are non-standard, so there are no parameters available in the existing force field. So we need to use some other ways of getting these parameters. If you are expert enough in understanding the ligand parameters, you can write them down manually one by one, or you can use some software. Now I'm going to use a software called ProDRG, which is an online server, to generate the parameters for Gromacs force field or Gromacs force field, especially, from which you will be able to get this parameters generated. Right. Okay, so here you when you search for the ProDRG, you can go to the run ProDRG, where you can give your ligand coordinates to generate the parameters based on that coordinates. Okay, now it is expected that your ligands are well minimized before you use it for this, or you can also run minimization here. So what we'll be doing now is that we will take our structure of the ligand, copy the entire contents of this. Especially, we only need the coordinate sections of this, the XYZ coordinates, not the entire way. But anyways, let's copy. You can paste your molecule here. You can request the tokens to be used. So I think my token is here. So this allows you to use 5 tokens at a time for your calculations. You can do 5 calculations. Otherwise, you will have to install it on your computer and use it. So I'm just going to use my token. It will ask you a few things like, do you want to make any changes to the chirality or check the chirality? Yes, it's good to check it. Do you want to use the full charges or the reduced charges? It's good to have the reduced charges. They will keep it as it is currently system. And whether do you want to do the energy minimization? Now, at this point, you don't have to do the minimization because we have already the dot complex, so it is minimized. Plus, if you run the energy minimization, it will change the coordinates in the GRO file that will be generated. So this software is going to generate the force field parameters for the ligand. Similarly, the topology file that we have seen, it is going to generate the GRO file, the GRO file of the ligand, which we cannot generate through PDB to GMX. So those necessary things, it will be provided to us by this particular software. And then you can simply run the software. It will take your molecule, check it, make sure that everything is good, and the outputs will be written on those you can download. So you can download all of these files as zip archive, and these will contain a lot of different files. I'll show you what it contains. So it contains the force field parameter files, the ITP files. It contains the GRO file of our structure, PDB file, the topology file, and many other things that can be useful for various purposes. Now, out of all of this file, we are only going to use 2 files. One is the ITP file, which is a force field file, and another is the GRO file, which we'll use as an input. So these 2 files, the drg.aph.gro and the ITP file, grg. So these are the files you can copy and keep in your folder. So these are the 2 we'll be using because ITP file will contain those things. If you remember from our topology file, if you remember, you will see that the same files are used for protein. The ITP file has been used for protein, for water, for ions. Now for ligands, we have generated this ITP file from the external server, that is the ProTRG. So we need to include that also over here. Now we have copied this file into the folder. Let's go back. Yeah. So we have the .gro file, which is coming from the server, and the ITP file are also there. So these contain the same type of atoms of the ligand which are there, and ITP will contain the force field parameters. Just to show you how the force field file looks like, it essentially has the type of atoms and its parameters, charges, masses, the bond angles, bond lengths, and its torsions and everything. So it will calculate the same parameters we already have for the proteins from the characteristic. Now this parameterization for different force fields, we have a different servers available. For CHARM, we have the FFCgen. For OPLS, we have the LigParGen, and these are different. I'll give you the list of all these softwares at the end, so you can always find appropriate software to generate the topology. Now, our topology of the ligand, and this is coming from the outside, so it is not yet included in our topology. So we need to make sure that it is included. So what we'll do now, we will update our topology file manually by opening it into the text editor. Any text editor you can use. This you will include. Now it is very important that you include the topology at the appropriate position. So we have not yet included the water molecules. We have not yet included the ions. So, but we are going to include first our ligand. So our topology file should come before water topology. Protein is of course there. So here you can just exactly same like this. Whatever statement you can see, the same statement you can put like include DRG GMX dot ITP. So when we mention this, that also during the run also include this parameter file or this kind of a force field file. So automatically the parameters will be taken from this file for the ligand. Next update we have to do is that we are going to combine the protein and ligand together manually. So we can use manual editing to include our molecule as well. So our name of the residue is K23, so that will come over here, and we only have a single ligand molecule, so that will be here. So this can be automatically set and the topology can be saved. Now we have saved the topology by adding some key terms, the ligand to ITP file and the K23 as the name of the ligand. Next, we need to combine these GRO files. So we already have generated the protein GRO file. Now we have got the ligand GRO file. So we need to combine these together so that we can use a single file for next all the processes. So let's open the GRO file of protein. Let's also open the GRO file of ligand. And it's very simple. Only thing what we'll do is we'll copy all these coordinates. Okay, you don't have to copy the last line or these first two lines, but only the coordinates you can just copy and paste into this file. Now remember here it shows some number which is called as the number of atoms in the current system. So here you can see that there are 910 atoms in the given protein. Here we have 46 atoms. So we are also going to change this number once we paste our atoms. So at the end, after the protein ends, we can paste the coordinates of the ligand. Sorry, I need to copy all of this. So only the coordinate section I'm copying from here, and I'm going to paste all of this after the protein ends. Okay, make sure the format and indentation is correct so that it all follows. Now we have added 46 new atoms to the file, so definitely we need to attach this number with this. So now this number will become 956, so that you will have your total number correct. Otherwise, you will get an error that the number of atoms is not matching in the current system. Okay, so let's take some questions. First, there are a few questions regarding the video. Yes, the video will be available on YouTube at exactly the same link that you have followed. So don't worry about it. You can revisit anytime. Kindly told Linux specification to PC. Yes, I will discuss that at the end. Which file is used in the ProDRG? The PDB file. The original PDB file of the ligand is which is being used in the ProDRG. So that is the coordinate file of your molecule. That's what we are using over here, and that's how we will get the output. Okay. So now that we have got to this far, I think we are all set to run few more steps of our simulation. So let's go ahead. Now, just to recap what we have done, we created the topology and Gromacs file of the protein by using the pdb to gmx command. We created the force field parameters and the Gromacs file of the ligand by the ProDRG server. And then we combined these files together by combining their coordinates. We also edited the topology file so that these new atoms which are added, they will be included in the topology as well. So at this point, you have successfully updated your topology and made sure that you have the correct configuration. Now we are ready to look through the next commands of the Gromacs and learn how we can set up next stages. The next command is going to be something called as edit conf or edit configuration, which will allow us to generate the box around the system that we have generated. So the command is very simple. It starts with, of course, gmx always. The next is the name of the program you want to use, which is edit conf in this scenario. It requires the Gromacs file of your system as an input. So whatever we have generated right now, the combined file I had combined is within the protein file. So that will be our input. So this file currently contains both the protein and the ligand. Next, it requires the name of the output file. How do you want to save the output file? So we're going to use the name md8box.gro so that we understand what exactly that file is. So this file is just going to contain what box, that's it, nothing else. Then what type of box you need, that is the bt command. So box types can be cubic, they can be triclinical, they can be octahedron, they can be spheres or shells. So you can use any one of them depending on that. The most common and simple to understand and process is the cubic. So let's use the cubic box. Next, there are a few more tags that we can attach. For example, C. So C tag is to center the molecule of the center of the box. If you don't use this, it will randomly place the box around the system. But when you use the C, it will make sure that the current system is at the center of the box. It will help in generating the periodic boundaries properly. The next, we can also specify the distance from the maximum distance from the edge of the minimum distance from edge of the protein to the box. So we can keep it, the units are in nanometer. So you can keep it 0.7 to 0.8 or maybe 1 nanometer to be safe. Now, where does these values come from? Are this arbitrary or does this really mean something? Well, when we discussed about when we discussed about the periodic boundary conditions, we saw that here, whenever you have these atoms, they are going to have a copy of them, the exact same image on their surroundings as well. And whenever we calculate these, long range electrostatic computations or interactions, we use a cutoff of around 10 or 10 angstroms or zero point 0.1 to 0.8 to so 0.08 to zero point 0.1 nanometers or so on. Now, if your distance between your protein and another image of the protein is very small, then we are going to unnecessarily compute the interactions within those proteins as well, which we don't want. So keeping the distance safe enough around 0.7 nanometers or 1, it will make sure that at least 1.4 nanometer, if it is 0.7, double the distance will be kept between the proteins. So between them, the interactions will not be computed. Our requirement to get the interactions within the given box only, the primary box, not in this box. So we don't want the cross boundary interactions. That's why the box size can be kept in this way. So for a safer side, I'm keeping it 1. If it is small, it will be, of course, better. It will be faster to compute. So let's enter this command. Just to repeat, the command is a part of the Gromacs program. The command name is editconf. It takes input, the system Gromacs file. Output is another Gromacs file. We can assign the box types, center the molecule, and also specify the distance from the edge of the molecule to the edge of the box. So once we enter, it will run the command. It will show you how you have run the command and what kind of system size. Now, what are the size of the box in all dimensions X, Y, Z? What is the volume of the box that is generated? So all of this see. So we have now edited something in the molecule. Let's always check. So every time we'll run the command, we're going to visualize what change exactly we are doing to the system. So I'll reload the new file that we just generated, which is called as the mdm underscore box dot grow. So when I load this particular file, and you will see, yeah. So as you can see now, currently our system has been put in a box. A box has been generated by this edit command. So within this, we are going to put our system, our solvent, our ions, our ligands, everything will be in this box. So it is just like a small test tube in which we are putting our solvent, we are putting our protein and all other things. So it's just like setting up a chemical reaction step by step. So this will set up the box around the molecule that we have, and the system is now ready for solvation. So the next stage in the molecular dynamics will be the solvation. To make sure that we are filling this entire box with the solvents, we are going to use the SPC solvents, but we can use many solvents like water or other types. Anything, any molecule you want can be a solvent. So let's go through the next command of solvation. In Gromacs, the command for solvation is called as solvate. The command names are pretty straightforward and self-explanatory. They tell you what exactly they do. So solvate is going to fill the box that we have created with a lot of water molecules. So let's generate this solvate command. So it contains few inputs. The first input it requires is the configuration of protein. That will be our box file that we just created. So this will be simply mdm box.gro. It contains configuration of solvent. Now this solvent configuration is already present. The GRO file for water is already present within the Gromacs. The name of the file is spc216.gr. So you just have to use it for SPC model. For tip 3, we have a different files within the Gromacs, so we can use this. Most importantly, from now onwards, every time we are going to make any change to the system, we have to always specify the topology file with this tag called P. So this P tag is always used for topology file within all these commands, and it is going to be always important. If you don't give this, the topology file will not be updated, and that will lead to a lot of errors at the end. Finally, we need an output file name. Let's call it MDM solvated.gr. So we know that this file refers to the solvated protein. And then let's enter. So as you can see, the volume of the system, the density that is currently being used, and as you can see, it's very close to the density of water. The total 8,303 water molecules have been added. Let's check that. Always make sure that to check. So first, let's check our topology file, whether it has been updated or not. So when I come to the end of the topology file, you can see this SOL after protein and K23. Now the SOL entry has been automatically added. Now SOL entry refers to the solvent. So you can see that 8303 molecules of the solvent are added. One protein, one ligand, and 8,000 plus solvents are added. So our topology is given. Let's also check it visually. Whether are this visible? Because we are using the explicit solvent, of course, these are going to be visible. So let's check that immediately in the VMD. So we can be sure that our process or the steps are actually working. I'm going to load the file that we just generated, that is mdmsolvated.gr. And once you load it, you can see here, now no more we just have the protein, but we have also the water molecules surrounding the entire protein. So these are all water molecules, as you can see. We are using the 3-point charges, simple point charges, but we have all the atoms, the hydrogens and oxygens and everything. So proper water configurations are used. So now our system is solvated. But that's not enough. Our system is currently at net charge, that is +5 charge we have seen already. Now, the life doesn't exist at a net charge. We always use buffers, salts in the buffers, and so on to make sure that the pH and the proper state is maintained so the system is neutralized. So the next stage is to use the command to ionize the system or neutralize the system, and then we can also check how we can add those. So slowly, we are building up our test tube for chemical reaction by adding different things by using this command. Now the next, this is just very early pre-processing. We still have to go through many steps, and don't worry, at every periodic, I will repeat everything that we have seen so we can go through slowly all the steps and again revisit the next one. Now, the next important aspect is adding charges or adding counter ions, especially. So our system charge was +5. So we can neutralize the charge by adding counter ions, which are negatively charged, and the total charge of those is 5. So you can add 5 chloride ions or some other negatively charged ions that can be also added. So let's see how to use the command. Now the command to run the ions is called as gen ion. But then the problem with the gen ion command is that if you go for GMX help and gen ion, it requires your structure, the TPR file, which we have not yet generated. We still have to go through the TPR file. So to first generate the TPR file, we used to use a preprocessor, that is GrowMPP, that we have used. Now for ions, I'm going to use a random molecular dynamics parameter file. You don't have to worry about it right now. I'll explain that once we start the minimization process. So the first command will be GMX GrowMPP, which will generate the necessary TPR files. Now for the GrowMPP to run, the required files are MDP file, that is your molecular dynamics parameters files. Here, they're not going to be very helpful, but they are just required for input generation. Then we need the configuration of our system, which is currently called as solvated.gro. We need the topology file because it is going to be updated, and we need the output file. Let's call this output file as ions.tpr. So we can have that. We can also provide the restraint file. Let's see if it is required. Yeah. Okay. We can also provide what was the I think, yeah, that will be it. We need a restraint file. Okay. So then we will use, it's giving some warnings about the force field because it is united atom force field. So I'll use the max one to silence those warnings and run it. So when I run it, it will prepare all the necessary files. So if you look at the folder now, the new file will be generated, which is called as TPR file, ions.tpr. So this file will contain the MDP file, the GRO file of the ions and everything together, which is used for the gen ion command. So after this, we'll compare. As you can see, the charge is currently 5. So we are going to use this information for adding charges. The next is the actual command to add the ion. So GMX gen ion. This is the command which adds the ions or generate the ions for the given system. The input required is the TPR structure, which we have just created, ions.tpr. Then the output, let's call it as MDM, the name of the protein, and ions dot GRO. So we know that this file contains the ions. So after creating the ions, this is going to be our actual full system used for minimization and step. So this is the last step during the system preparation. Then we have to give the topology file always so that the ions which are added can be added to the topology. Next, we have to give which positively charged ion we want to use with the tag called pname. So pname is for which positively charged ion. So we can give like Na, K, Ca, and any other things, calcium, magnesium, whatever you want. Similarly, you can also give what negative ions you want to use. You can give like Cl or other things. After this, you can mention how many ions you want to use. So whether you want to use, let's say, for example, NP is number of positive ions. So how many you want? Of course, we want 0 in this case because we already have positive charge. How many negative ions you want? You can give this way. Let's say 5. So we need 5 negative ions. So what it will do, it will take the chloride ions as the ions, their structures, and 5 of them will be added to the system. So that can be done. Or there is another simple way or shortcut. You can simply say that neutral. So it will automatically calculate the charge and make the system neutral by adding the necessary charges out of these, whatever will be required. So once entered, it will ask that currently the box is filled with, completely filled with molecules. So which molecules you are free to replace, because to add the ions, we have to replace something, right, in the box. So that is going to be water or solvent molecules. So solvent group is automatically generated here, group number 15. So you can use the group number 15 to replace. And once you enter, you can see that solvent molecules, the 5 solvent molecules have been replaced with the Cl ion. The ions have been added. That means now water anyways will be the neutral in their molecules, so it's okay to replace them. So after replacing the water molecules with Cl, our total charge on the system will be neutral. This is very important, especially when we use the long-range electrostatic calculations. So it has to be performed for this. All right. Now, at this point, we have the system prepared. Let's, as usual, first check the topology file. At the end of the file, you can see now the new entry has been added called Cl, and 5 of them has been added. So automatically these will be given. The force field for Cl, SOL will come from these ions.itp, water.itp, spc.itp, and for our ligand, we have added the drg-gmx.itp externally. So this way we can make sure that everything is properly set into the simulation. Let's also check the system visually, where exactly those ions are and are they actually present or not, just to make sure that everything works fine. So let's load the system that we have just prepared by using these different commands. Our file name is mdm-ions.gro. This is our final system we're going to use. Let me change the graphics a little bit so that we can understand each part differently. So first, let's look at the protein as the ribbon representation. Let's look at our ligand, and let's call it as stick representation. So you can see the protein and ligand so far. Let's add our ions and show them as spheres or van der Waals spheres. So you can see these are the 5 atoms of chlorine which are used in the system. And of course, finally, we have our water molecule, which are the part of the system anyway. So this is the entire system we have prepared. So now this system is ready to go for the further stages of simulation. Okay. So we have prepared our system by using this set of commands. We can also prepare systems simply by using some other tools like UCSF Chimera, where you just have to click on the commands like add H or solvate or add ions. It will automatically do this for you, but it may not always be compatible with all the force fields we are going to use. So it's better to prepare it from the scratch by using the appropriate force fields that we are using, so that it can be processed without any errors. Okay. So now just to recap, we essentially prepared our system by using a series of commands. These commands from the top were first the PDB to GMX, which helped us generate the topology, the restraint file, and the Gromacs file of the protein. Then we use the topology file of the ligand from another server, that is the ProDRG server, and combined them together by updating the topology and updating the Gro file. The next command was the edit point, which generated the box around the system of certain size and certain shape. After generating the box, we use the solvate command to fill the box with water molecules by using the SPC model. After solvating, it was not still at a net charge, so we use these two commands, GrowMPP and GenIon, to generate the uh, ions. So this GrowMPP command, it uses the MDP file. We'll now learn how does the MDP file looks like and what each of these parameters actually mean just now. So now onwards, every step will be like this. First, we will run GrowMPP and then the MD run. GrowMPP, then MD run. GrowMPP, then MD run. Because this is the preprocessor for every MD run command. So this GrowMPP uses the parameters and other files like topology and GR file to generate the TPR file, which is required by many other commands like GenIon or MDRun. After generating the TPR file, we run the GenIon file to generate the ions, which can neutralize the system based on the charges and give us the complete system ready for further stages. Okay. So at this point, we have created a very good system and looks good to go. So from here onwards, we'll start our next stages. So if you remember, the first stage in the simulation was minimization. We need to minimize the system so that we can properly set the base for starting the equilibration and further steps. So it doesn't have any unnecessary steric clashes or steric strains within any atoms. Now, for every of this, we are going to need the MDP file as I discussed. So first is the energy minimization. For that, I have created a file called em.mdp. Let's open the file and see what are the options or parameters provided here. So these are very simple basic parameters that I have added, but there are many others available. So there are parameters like which integrator you want to use. Now integrator means which algorithm, whether you want to use steepest descent. So steep is for steepest descent. If you write CG, it will run conjugate gradient. So these two are for minimization. If you use the algorithms like MD, it is for running actual molecular dynamics. So whenever you want to use the heating or equilibration or production, we use the MD algorithm. And for minimization, we use the steepest descent or conjugate gradient algorithm. Then the next parameter EMTOL is a tolerance for energy minimization. It means at what force constant you should stop the minimizations. So that is by default 1000, which is good enough for most common systems. So if the force becomes lesser than that, it will stop the minimization process. Or if it doesn't happen, the minimization will be stopped with the NS steps or number of steps. So total number given currently here 50,000, which is good enough for the average site system. And your minimization is of course going to stop even before these steps are reached. But it's good to keep a upper limit for these systems. And the energy minimization step, like how often they should check the energy minimization calculations. Then the parameters which refer to different types of calculations of interactions, the cutoff schemes that we need to read, how do you want to save the neighbor list, and so on. What kind of Coulomb type interactions need to be used. So most common is the particle mesh evolved, which is used to treat the long range electrostatic interactions properly. Then for Coulombic interactions, what should be the cutoff? Basically, this cutoff means that after this distance, if the two atoms, non-bonded atoms, are apart more than this distance, then the Coulombic force will not be generated or calculated. The reason, if you remember that graph I showed, during the very small distances, the Coulombic forces are very dominant, but as and when the distance grows, the forces become very low and negligible. So we can give a cutoff, so our calculations will be reduced over here. You can change it to even lower, like 0.8 or 1 also, but this is good enough for common calculations. And if whether you want to use a switch or direct cutoff and so on. So it will force switch means it will slowly, gradually change from 1 to 1.2. It will not just cut directly. So it will try to gradually reduce the interactions which are being calculated. Then the long range van der Waals cutoff. So same cutoff can be used for different long range interactions. Do you want to use the PBC or periodic boundary? Yes, we are using periodic boundary in all the three systems, that is X, Y, Z, all the three directions. So 26 boxes will be generated. I will show you how do they look like. And dispersion correlation, whether you want to calculate. Right now, it's not important because we are just running minimization. For pressure related calculations, this may be required. So this is the input parameter file or the MDP file of the energy minimization. Let's run the energy minimization and see what happens. Okay. So the first step always is going to be running the grow MPP command, which will combine all of these things: the GRO file, the TPR file, and the GRO file, the topology file, and the MDP file together, and that can be then used for further MD run or actual minimization. So let's minimize the system. So GMX grow MPP. Then the input will be the MDP file that we have created. Then now for beginners, these MDP files, you can use the sample files from online or the database and just change the values. But you can also from the scratch write them. If you are remembering all the parameters, you can also write them from the scratch. Now, the next thing it requires is the Gromacs file of our system. So our system, the final system is called as mdmions.gro. So it contains everything: the protein, ligand, water, and the ions. So everything is present in this file. So that will be our initial file for minimization. Then the topology file. So whatever parameters will be updated, they will be saved into the topology file. Then how do you want to save the output? Here the output will be em.tpr. So tpr file will be generated by using the grow mpp command, and you can then run it. Okay, I have to use the max one for the Gromacs force field just to silence those warnings. Okay, so now once I run the grow mpp, the tpr file will be generated. It will also suggest what's exactly happening. It will analyze if there are any errors, anything wrong, it will show you here. So there are three types of things which are given. First is note, second is warning, and third is error. Notes are just information that you can take as a suggestion. Warnings are things that could go wrong, and errors are often fatal, and with error you cannot go ahead. But with notes and warnings, you can still go ahead if you feel that the warning are harmless. But if they are error, you cannot go ahead. And it will be explained exactly what it does. So now we have created the TPR file. Actually, now run the energy minimization. To run the minimization or any MD algorithm, we have the command called mdrun. So this is going to be now repeated: grow mpp mdrun for every step. It requires the input, that is a TPR file, and the names of the output files. But easily there is an option called def nm present over here, which can automatically use the same nomenclature for input and output files. So when I say that the def name em, that means it will use the em.tpr automatically, and all the files generated will also have the prefix em. So we can easily make out which files are from the which run. So, and we can also use the verbose command to just show what exactly is happening on the system. So let's run the mdm command and our minimization will start. So as you can see, the minimization steps have started, and you can see these are going on. The energy is becoming less and lesser. Finally, when the force constant is reached to the threshold that we have given, it will stop minimization. Or if it doesn't reach, it will go till the 50,000 steps and then it will stop. So now the energy of the system is something like this: negative 3.9 into 10⁵, and the forces on everything which are on the system. So now we have minimized. Let's check this Gromacs file and let's see what exactly is happening in that. So our system actually has minimized. The files generated will be em.log, which will contain the log, em.gro, the Gromacs file after the minimization, the new coordinates after the minimization, the TRR file, which is the actual trajectory of the minimization. So that is there. So our system is now minimized. If you check the energy file, the EDR file will also be generated, which will contain the energy of different time points at every step. Now remember that the energy minimization is not time dependent; it's a stepwise calculation. So one by one steps will be calculated without any time constraints. Our system is now well minimized. You can also check that by plotting the graph of energy and see whether it has been minimized or not. But now our system is ready to go for the next stage, which is the heating and equilibration and actually preparing the parameters. So this is where the things are going to get now a little bit complex because of the presence of the ligand, everything that we have to do, a lot of things manually, edit files and generate a lot of parameters for this purpose. So let's start talking about different aspects of this preparation and how to do that. Okay, so before I go ahead, I know that for beginners, this could be a lot of things. Are there any questions or any part of this which is not clear? Please let me know. This is not a stuff that you can grasp in just one go. You might have to practice it one more time by yourself. I'll share the sample material, the files, and the commands, the list of all the commands written documentation with all of you in the description of the video later. So you can check that out and just try to run these commands on your own. So you will get more confidence. Okay. So there is a question about the glycam force field. Yeah, so if you want to import the glycam force field, you can simply go to the glycam website and download the force field, which is compatible with Gromacs. They have different files. And just put the force field file, the FF folder, into your data, wherever your data or from wherever you are running the command. So whenever you have, it will automatically detect that force field. So whenever you run the PDB to GMX, it will also show you that force field in the list which is present in the current folder. So you can directly use it. Okay. Now we have minimized the system. So we are gonna start looking at the molecules and the properties and create some detailed groups to track our atoms of different systems into this and prepare ligand properly. So the next stages, before we go for the heating and equilibration, we have to prepare two things. First of all, we have to generate the restraints on the ligand. The protein restraints have already been automatically generated. So if you remember from the very first command that we ran, that is the PDB to GMX, it generated a file called posra.itp. Now this is automatically generated because these parameters are available. But for ligand, we don't have such files. So we need to generate this file. Now, first of all, what are restraints and why they are required? So restraints are the kind of limitations on the movements of the atom which are required initially during the equilibration box. Because if you directly run the MD without restraints, it will break apart the molecules, it will break the simulation and just fail it at some point. Because the system is not yet at the equilibrium, close to the equilibrium, the heating, the temperature we are applying, the pressure we are applying, it may be too much for the system. The forces generated will be too large, so it will simply break apart. So it's important that in initial stages of equilibration, you keep your main system, the protein and ligand, restrained at the same position and just allow them to have a very small or minimum vibrations in the local area, not proper like free. So we need to generate these kind of restraint files for the ligands and make sure that everything is included in the topology and editing as well. So we are going to learn few more commands of the Gromacs which are extremely useful. The first command is called as index groups. How to generate index groups. So index groups are extremely interesting part of the Gromacs. These are the group of atoms you can generate and keep track of them or monitor them for many different purposes. So if you remember some of the earlier commands that we have run, it was asking us to choose systems. So as you can see, for the given system, Gromacs automatically generates the necessary groups. So group 0 means the complete system. Group 1 means the protein, only the protein part of the system. Group 2 consists of all the atoms of protein but without hydrogens. Group 3 contains only the C alpha atoms of the protein. Group 4 contains only the backbone atoms of the protein. Group 5 is main chain. Here we have main chain and carbon beta, main chain plus hydrogens, and so many others. As you know, even if you, whenever you added that K23 ligand, it is actually a other residue or other type of ligand. So it will come under two things, other group, and the K23, its own group will also be generated. We have another group called water, another group called solvent, in case solvent is other than water, and non-water. So rest, except water, rest of the things which are there. So these are automated groups. Now, whenever we're going to put the restraints, we can directly use this K23. But the problem is that when we put the restraints are only put on the main chain, the main atoms or heavy atoms, not on the hydrogens. We cannot put restraints on the hydrogens because hydrogens are extremely vibrational. Their frequency is very high. So they cannot be directly coupled with rest of the things. They have to be taken care by some other algorithm. So we use like algorithms like links, or we use shake or rattle. These are the algorithms which track the movements of the hydrogens and control them during the simulation. So we need to create another group which will only contain the atoms of the K23 ligand which are not hydrogens. So without hydrogen, the rest of the things we need. So we need to create our new group which will be then used along with the rest of the parameters. So and this way you can have any group. Okay, if you want to track, let's say bindings and residues for energy, you can always generate that group of those residues and track it and across the entire simulation. So their energies will be reported separately. You can do a lot of things with these groups that you generate. So it's a really good thing to use. So let's see how to generate a group of the ligand with only main atoms and not the hydrogens. So for the command to make the groups is called as make NDX. It is the part of the Gromacs again. This can be used to generate any type of group. For which file you want to use the group. So we give basically the .gro file as an input. Now our file was drg drgaph.grow only for ligand. We don't want protein to be part of this. That's why we are using only the ligand file here. And then the output. So let's call this group as index group for K23. So we know that what kind of group it is, and often the extension is NDX. So when we will enter this command .make NDX for the ligand, it will ask you what exactly you want to save in a group. So here you can see that 0 can be automatically 3 groups can be made, a system, other, and K23. Do you want to make anything else other than that? Well, you can use any one of these combination. These are all Boolean operators, and at this point you don't have to strain yourself much with this. Let's just go ahead. So here we have different options to select different types of atoms from the given system. So you can use the specific group, you can use the specific type of atoms, you can have the atom types specified. You can also combine different atom sets by using and or not type of logical operators and so on. So let's see how do you want to create a group by using this combine. First, let's say, let's type 0. 0 means use the entire system. Then we combine this with another condition. So let's put and, it will put together two conditions. And the second condition we have to give is not hydrogens from the system. So not. So for not, we have the symbol called this exclamation mark. So let's put the exclamation. Then for atom type, we have the symbol A, so that we can put. And which atom type we want H, or let's call it H star because they will be numbered. So this entire statement here states that as long as the atoms are part of the system but not hydrogen, take them into the group. So it will automatically create such group. And when I enter, it will generate a new group called system and not hydrogen. So it will automatically generate such group which will consist of 32 atoms. So out of 46, 32 atoms groups has been. Now these atoms we can restrain, but if you would have used all of the atoms for resistance, it would have been very unstable system, and the forces would have broken the hydrogen bonds and unstabilized it. So now we can save this group by using the Q and just enter. So the new index group will be formed. So if you check your folder, the new file named as index K23NDX has been automatically formed, and rest of the groups are inbuilt into the Gromacs. So now we can also track these groups for different purposes. Now let's create the restraint file for the ligand as well, just the way we have earlier done for the protein. The command is very simple: gmx gen restr, that is gen restraints, generate restraints. It takes your input GRO file, the same we have used to generate the groups. So gmx aph, sorry, this is drg aph dot gro, that will be our GRO file for the ligand. Then it requires the index, like which atom you want to generate resistance for. So we just created the index. So what it will do, only for those atoms in the index, it will generate the resistance on this molecule, and others it won't. Hydrogens it won't. Then you can give the output name. Let's call this as position resistance for the ligand, that is post re underscore K23 dot, the file extension is ITP. So these are the force field parameters only, but at the same location. So post re K23 ITP, let's call it. We also have to give some force constant that needs to be used for resistance, like how much the atoms are allowed to move, how much resistance to put. Again, the force constant can be exactly same that we used earlier, that is 1000 kilojoules per mole per whatever nanometer or what. So this can be given as in each direction. So this will be the force constant. So as long as the force constant is maintained, the movements will be allowed. But if the force constant goes beyond that, below that, it will stop movement. It will restrict the movements over there. So let's enter. It will ask which group you want to use. Of course, this newly created group, we want to use the group number 3 and enter. So now you can see, again, check the folder. A new file will be generated called post-re. So these are the positional restraints for the ligand. This is going to keep the ligand at the stable position during the initial stages of equilibration till we reach to the equilibrated system. So that's what it will do. Now, after this, we need to include all of this information into the file, that is the topology file, because this file needs to be going to the topology file. So let's open our topology file and start editing it. Okay, so we have a lot of these different things over here. So as you can see here, how the restraints are applied. So you can see here the restraints for the protein are included as if def post res include post re.itp, which was created by 3db gmx and nd. That means that if the simulation requires positional restraints, only then use this file. Otherwise, don't use it. Same thing we have to now give for the ligand. So for restraints are water are already included, ions are already included. Now all we have to do is include the restraints for our ligand. So just after our addition of the ITP, we can include that. So the statement goes like this: if def POSRES. Now this POSRES means that this definition we include in our molecular dynamics. Wherever, for whatever run we want to apply the restraints, we have to give this definition at the start of the file so that it understands that we want to apply the restraints, and only then it will be using this file. In the final production run, we are not going to apply any restraints. The system is going to be completely free. So that time it will not use this restraint file at all. So if def POSRES include the file that we just generated. The name of the file is POSRE underscore K23.itp. So whenever in the system this keyword will be used, automatically this file will be used, and after this end if, so that if condition will be stopped. These conditions will make sure that only at the required points it is used, and then you can save this topology file. So our restraints for the ligand are also now done. Now, before we start the next quick runs of this, one final step of the preparation is left for the ligand to be properly handled. So before equilibration, let's check what kind of files do we need. So let's go for our input parameters. Now we are going to heat the system, then equilibrate it. Heating, equilibration are both just running MD. Only thing is these are restrained. We are going to restrain the proteins and ligands in place and couple them so that they can be tracked later and stabilized. And final MD will be unrestrained. You don't apply any restraints. So first will be the heating stage, and heating we are going to do with the NVT ensemble. That is the number of particles, the volume of the system, and the temperature will be constant, and the pressure will be allowed to change over the time. So it will heat the system by applying different pressure and make sure that all the atoms are at the required temperature, the kinetic energies and everything is good. So the input parameter file for the purpose of this looks something like this. Okay, before that, there is one more question asked: how to set force field for calcium or magnesium? For calcium, magnesium, in most force fields, these molecules, these ions are already included. Like charm, you can use, or Gromacs, you can use. So already there are. You don't have to add any separate force field for these. Most common metal ions also, you don't have to use anything. All right. Okay. So now if you look at this file, it contains few more parameters than the energy minimization. It contains actually many. So let's go through some of the most important and understand how to set this thing. So first is the define. This is what we just discussed about the POS RES, that is the restraints. So this define uses the tag called DPOS RES, that is positional restraint on the proteins and ligands, whatever the files are included. So now for this simulation or this NVT run, it is going to use the restraint file that we generated. Which integrator? Now onwards, for every step, the integrator will be MD, the actual leapfrog integrator. How many number of steps you want to use, and what should be the time steps? Now this is very important to show. It depends on system to system. So I'm going to use a very small number of steps to show you quick results. So what here it means is that time step is like, so all of the units for these are, remember, in picoseconds. So time step, that is dt of 0.002, means that this is the 2 femtosecond time step. So every 2 femtosecond, the calculations will be performed by the integrator. And if you multiply this with the number of total steps, you will see that this will be total of 50 picosecond of simulations that will be generated. So we are going to heat the system for 50 picosecond, and that is the total time for. Of course, you can increase. It's better to heat it for at least some time and then equilibrate for more, at least a nanosecond or two, and then final production run, you can have any time scale that you want. So these number of steps and the time steps will decide how long your simulation is going to run. So this simulation is going to run for around 50,000, like every 2 femtosecond is going to calculate into the 25,000 total steps. So the actual run it will be produced will be of 50 picosecond. All right. So that will be recorded. If you want to increase the time, you can always increase the number of steps from here. Okay, let's do 100 picosecond. So let's just double this number to 50, and the total system is going to be around the 100,000 steps or 100 picoseconds now. All right. Now, in this, all the file, as you can get the pattern very clear, the variable name is on the left hand side, the value on the right hand side, and there are some things like, followed by semicolon. So these semicolons are called as comments. These are not interpreted by the Gromacs. They are just for our understanding. So you can write anything in these comments. It doesn't matter. These are just for our understanding. So we have essentially set up our total time for the heating phase. Then the output control. That means how often you want to record the output. The thing is that you can, of course, record every step, but it will generate a very huge amount of data. That's the problem here. So we don't record every step. So we record the data every few steps. So for example, here, as I can show you, the NST energy, that means how often you want to write the energy output. So I have used 500 here. That means out of these 100,000 or 50,000 steps, every 500 steps, you save the energy data. So it will save the energy every picosecond. Then NST log, how often you want to write to the log file? Again, the same. You can always increase or decrease the frequency. If you have a lot of space on your computer, you can write it more often. If you have less space, you can write it infrequently. Then how many times you want to output the trajectory? Okay, so either you can use NST out, the coordinates, which will generate the normal trajectory, or compressed, which will use the compressed trajectory. So save the coordinates of the current state every 1 picosecond. So this will create the output parameterization. Then the bond parameters, like how do you want to run? Are you continuing the actual earlier MD? Well, of course, no. This is our first step towards the MD. So this will be no which constant algorithm are used. So as you can see, I'm using here links, which is a very good with respect to these force fields. And the constants used are only for H bonds. So all the bonds containing hydrogens will be constrained by links. And rest of the restraints are anyways we have in our positional restraints. Then going forward, we have many other things. The parameters for the links algorithm, the same thing we saw earlier, the neighbor searching and van der Waals and electrostatic cutoffs and all those stuff will be remaining same. The new things you will see is that temperature coupling. Now we want to heat the system, so we have to provide a temperature coupling. So there are various algorithms for temperature coupling. One of the most commonly used is V rescale or velocity rescaling. It will keep the temperature constant. This is called as thermostat. And this will keep the temperature constant during the simulation by providing or reducing the kinetic energies of the system. So velocities will be rescaled as per the temperature drops or increases in the system. The temperature coupling groups, this is very important. How do you want to couple the temperature on the system? So you can use different groups, whatever you want. So currently we are using two groups here, protein K23, but we don't have this group right now. We need to generate this group. So we are going to generate it after that. And another group is water and ions. What exactly is happening here is that the protein and ligand are going to be coupled to the temperature together. So their temperatures, their movements will be tracked together. If you use a individual coupling, like for ligands separately, ligands are very small molecule and it will be very unstable. Such a temperature will break, it will be unstable and it will break the ligand. So you should always couple the protein and ligand together by creating their group. And water and ions, the rest of the things can be auto. Now this group is automatically created. We'll see that as well. So each one of the group is like coupled with the coupler to, one by one, and it will make sure that they are tracked individually. This is the time constant, like how often you want to check for the temperatures and everything. Then the reference temperature. So we are using 300 Kelvin for both of the systems. Of course, the entire system will be the same. So you can change this temperature to anything that you want to test. Now, as we are doing this heating at the NVT ensemble, we are not using any pressure coupling. So it's no. The periodic boundary conditions, of course, we are going to use always the XYZ. Now, the dispersion correlation, again, we don't want it here. It will be only required with the additive force field, which are different. Velocity generation. Do you want to generate the velocity? So in the first step of MD, it will always be yes. This is just the first step. So we will generate the new velocities. But when after this, every time it will be no, because it will be using the velocities generated by this earlier run only. Then the temperature, of course, it will be as same as the reference temperature, and the random seed, if you want to put, it's optional. So this MD parameter file is very detailed for the this kind of heating, but let's run it anyways. So let me save the change, that is the time, and now let's run. But in the MDP file, if you remember, I have given a new group for which I don't have any new group available. That is the protein underscore K23. So how do you generate a new group which consists of both proteins as well as the ligands? So again, we can use the same command, very simple, gmx make ndx. Then your input file will be your current system, that is em.gro, which we're going to use for the final next step. And output will be the new index file, that is index Okay, let's call this new file as index.ndx simply. When you go for this, it will ask you which currently available systems in the molecule you want to use for this. So we're going to use protein plus K23. So both of them we want into the same group. So new water enhance, as you can see, automatically created group and all. So for protein, the number is 1. So let's press 1. For OR, we have this bar. So we will use OR and K23, that is number 19. That means that as long as atoms belong to either this group 1 or this group 19, so any of the condition is true, you add it to the new group. So automatically protein underscore K23 group will be generated, and that's what we are coupling for the temperature into the heating. So let's enter. The new group number 22 has been generated. The name is protein K23. You have to make sure that this name is exactly same as what this is the exact name you want to write in your MDP file also. So after this, we can save the group and then close it. Now onwards, this new index file or new group that we have generated, we are always going to use it with all the simulation. This is where we are now ready to run all the stages of simulation quickly. So now it won't take much time. We are just going to run each command quickly and see how does it work. So first will be GMX. So we are going to start running the MD run. The first will be heating stage. So let's start with the GMX grow MPP first. The file here will be nvt.mdp, the molecular dynamics parameter. The configuration will be minimized configuration, that is em.gro. The restraints file will be the exactly same for now that we're going to use, the em.gro. So by using that as a reference, it can restrain the structure. Topology file is always required because we want to update it. And the new index we have generated so that it can be tracked. So the index file name, index.ndx. And finally, the output file, which let's call it nvt.tpi, because we are generating the nvt run. That is the heating phase. And let's enter. Okay. So I have to again forgot to use the max one for Gromacs. Okay. So now you can see that this run is gonna roughly generate around 50 MB of data, which is good because I'm not saving the coordinates very often. Just one by one, I'm saving every 1 picosecond. So now our grow MPP is finished. Now it's time to run actual heating. So let's start with GMX MD run. Then the def name. Now in this case, our TPR file is called as NVT. So let's use the same name even for the output files. And let's keep the output verbose so we see what exactly is going on. So this run will heat the system and it will start heating. And you can see now the different steps are going on. We have given around 50,000 steps. So it will take some time to finish all the steps, maybe a few minutes, depending on your the processor and the GPU and the machine configuration. It might be fast or slow. So you can just run it. So I'm running this on a laptop, so it may not give a huge performance as such for the system, but it is using the GPU, especially for the PME calculations, that is the particle mesh evolved long range electrostatics, and the 16 cores of the CPU it is going to use for the rest of the calculations. That 8 OpenMP threads have been generated on CPU. Okay, so once this finishes, our heating stage will be done. And yeah, we can immediately check whether our heating is finished or not, or what has happened to the heating, all the stuff. Let's check that as well quickly. After it is finished, we will check. All right. So, all right. Now our simulation has finished. It will the heating phase has finished. It will show you some parameters like at the end, what are the things, how long did it take on a computer time, and what is the performance with the current system. So as you can see, my system is not giving a very good performance, but for this small system, it is providing 120 nanoseconds per day. So if I want or 0.195 hours per nanosecond. So if I want like 100 nanosecond simulation, it will take 19 hours, 19.5 hours on my system for this protein. If the protein size is big, of course, it's going to take much more time. This is a very small protein, just for an example. So we are using. So our this has been properly heated up. Let's check the graph or the energies or whatever we have so far in this. So we can also check the energy. We can also check the heat, the temperature, and so on. So let's first check the energy from the energy minimization, and then we'll check the temperature from the heating. So again, for checking that intermittently will be a good idea just to monitor so that you are sure your system is running very well and there are no problems in the simulation. So how do we get these energies and so on? So there is a very simple program called energy, which is a part of the Gromacs, which can be used. So we just have to give GMX energy, then the energy file. So for energy minimization, the file was em.edr. Then for actually giving the output, you can say energy.xvg. It will ask you out of all these parameters which you want to plot. So first, let's check during the energy minimization whether the energy has been minimized or not. So the potential energy is what we are looking for, number 10. So you can put number 10 and 0 for the time scale. And then you can see the file will be written. And this file you can always check with the help of plotting softwares like XMGRESS, which will show you whether during the minimization what happened. So if you see the energy has actually minimized, it's not visible because the graph is very sharp. So there's a very sharp decrease. You can see this line here in the energy and it has come to the minimum. Now let's check the same thing for the temperature, whether the heating has changed anything in a temperature. It may not have much changes because we have run it for a very small time. So GMX energy command will be same. The file will be nvt.edr and the output will be temperature.xpg. It will ask what you want to plot. Again, here we can select temperature. That is number 15 and 0 for the time scale. It will produce the file. Again, you can plot it just to check how your simulation is. It's a good idea always to check. Now here, if you see the temperature graph, it looks like that. But yeah, overall the temperature has been maintained. It's fluctuating over the time, but it has been maintained around 300. The fluctuations are not too big. Maybe plus or minus 2 degrees is okay for now. Maybe you can allow it to heat for a little bit more time so that everything becomes a little bit stable at this temperature. So our system is now heated. Now we have to go for the equilibration. So let's check the equilibration. We are going to perform the equilibration at the NPT ensemble. That is, number of particles, pressure, and temperature is constant. So pressure is not going to change. Volume free in the sense the system is now free to expand or contract depending on what interaction it has. So it can be kept free. So here, as you can see, the things would remain same. I'll increase this to little bit more. So let's instead of 25, let's keep it 100. So this will be the 4 times. So this will also be 100, and this will be around 200 picosecond equilibration. A good equilibration is always good. It doesn't harm. It will properly equilibrate the system. The pressure and the density will be nicely equilibrated. Now, the only difference we will see here is that earlier our pressure coupling was off, but now we have to keep the pressure constant. So the pressure coupling is on, and we can specify which coupler or which barostat you want to use for this purpose. So all of this can be given. Again, these references you can find in the documentation, that what are the names of different algorithms and everything, and you can put. You can see that now the generate velocity is no, because we are going to use the velocity from the earlier run, whatever we had. Whether it's a continuation, yes, because we are continuing the simulation from the earlier, so it has to be used as continuation from NPT. So yes, so these are the only thing. Rest of the things I'm keeping the same, not really changing anything. Now it's very simple. Just run the same command but with the new files. So we will start with first the grow MPP, which will be giving us the necessary TPR file. So GMX grow MPP. The input file will always be the MDP file. So in this case, we are using NPT as our file. The configuration file will be the output of earlier command, that is nvt.gro. The same will also be the restraint file for this, that is nvt. .gro. Even here we are going to use restraints. Only in final run we are not going to use restraints. So this also requires few more things, like we can also give topology file. It's always required, very important. And finally the output file, which will be called as npt.tpr. Oh yes, and most index file also, because we are tracking a new index for coupling, so we can also give the index file that we just created. With this, we are ready to generate the TPR file for the NPT equilibration run. So once you enter, why I'm always forgetting. Okay, so this will run the simulation and generate roughly around 28 MB of data. This is good enough, not something big. Now we'll run the equilibration. So we are gonna issue the command gmx md run. So as you can see the pattern, grow MPP and md run are just coupled. Just the files are going to change all the time. And the def name we are gonna use is npt here, because our files will be npt, and then start running. So to start running, it's gonna use 100,000 or 1 million steps. It will be around 200 picoseconds, and it will run for those many steps. So it's gonna take some time, around 140 seconds, 2 minutes plus something, based on this PC configuration. And once that, our system will be well equilibrated. During that time, you can see that a lot of different files are being generated. So for NVT, same as energy minimization, we have all the files. We have the XTC, that is the compressed trajectory. We have the checkpoint file, just in case this simulation stops, you can use this checkpoint file to continue. The energy ADR file, the GRO file, the log file, all of these are generated. Same goes for NPT. So every run, it will generate all of these different files, which you can use for further analysis. So yeah, just 1 minute more, and then we can go for the final production run, and our simulation will be finished. Let me check. Meanwhile, are there any questions? All right, no. Yeah. Finally, once this production run is also over, now we will be able to visualize the trajectory, see the video or animation of how exactly the atoms are moving, what are their movements over the time, and perform the analysis. So once we perform the final MD, we will transform the trajectories and start analyzing it for various different types of data points that we need to report our MD results. So it's almost finished. The final run is just going to take some time. So we might have to wait for that. But anyways, meanwhile, we'll be discussing the trajectory transformations and so on. Okay. So our equilibration is completed. So you can see some basic parameters that it has generated. You can see the performance of your final steps. It's giving the same performance, around 126 nanosecond per day for this system on this configuration. It may be different for every system. So now let's check whether our configuration, how it has done the equilibration. We can plot the graphs of our density of the system, the pressure on the system, just by using the same command. So gmx energy, it will always be the thing. The input file will be our EDR file, that is npt.edr. And then output file will be, first let's try to put pressure, plot pressure.xvg. So pressure is at 16 number, so let's use it. And then by using plotting tools like xmgress, you can simply generate this plot. So you can see that the pressure has been fluctuating in the system. So system needs to be still equilibrated more than this, but it is around like 1 barometer it will try to keep. Okay, but it has to be more equilibrated to be able to stabilize before MD. For now, I'm just going to use it directly. It's a very small system, so it may not have any artifacts as such. The same way, we can generate the graph for our density as well. What is the density of the system and how it has been maintained throughout or not. So if there are any thing wrong going in your system, any unnecessary forces generated, any pressures is increasing, you will be able to see that through the plot, and then you can rerun with some more parameters before you go for production MD. So it's good idea to check these parameters periodically to make sure everything is in place. So the density as 22 number, so I'm going to plot it. And then you can get this also plotted by using the XMGlass and see the density of the system. Okay. Yeah. So initially it was used, but with the solute being dissolved now into the water, the density might be different. So accordingly, you can see the density has been changed over time. So that is also fine. So our equilibration is done. Now we are ready to go for the final molecular dynamics, the production run, what we call as, which is what we're going to use for all the analysis, all the thermodynamic equilibrium calculations and ensembles. That is what we're going to use. So far it's just been preparation of the system for MD. So let's have a quick look at the MD parameterization, that is md.mdp file. Now here, the first difference you will observe is that there is no more positional restraints. Now the restraints are not going to be used. The system is absolutely free to move as per its own forces and interactions that it generates. I'm running it for 3 nanoseconds, so it is going to take some time, but let's see how long it takes. Then maybe it take around 10 minutes or so, 10-12 minutes. Let's see. The time step is just 2 femtosecond, or maybe we can reduce it a little bit. Let's make it around 2 nanoseconds, just for an example. So this will be 2,000 picosecond, 2 nanosecond. Rest will be the same. The saving and all, we are going to do it every 10 picosecond so that we can save on some data and it will be speedy. And yeah, so rest of the things will remain the same. We don't have to change anything here. Only thing, we have now removed the positional restraints, so we can keep it system completely free. So to run, again, it will come to the same thing, GMX grow MPP, which will be preprocessor. It will take our initial inputs. The parameter will be F. The MDP file will be the first parameter required for the grow MPP. The next will be the checkpoint we don't have to keep, but the configuration file, which will be the output of the earlier command, that is npt.gro. Then the topology file. Now here we don't have to give the restraint file that we gave earlier because we are no more keeping the restraints. We are just keeping it free. But we have the temperature coupling group, so we can give that index.ndx. And what should be the output? So let's call this output as md of ligand dot TPR. So it will generate the TPR. So let's enter. Okay. I'm just really forgetting this. Okay. So again it will generate some data and it is good enough. Now we will run the actual MD. So GMX, then MD run. It will take input, that is the def nm. We have given is MD of ligand. So let's keep that as a name for output as well. And let's keep the output verbose. If we don't give this V, it will run, but silently. It won't show any output while it is running. But it's good if you can see what's happening. So let's enter. So it will start the final production MD. It is going to run for 2000 picoseconds or 2 nanoseconds. And it's going to take some time, as you can see here. Oh. Okay. It's actually going to take a lot of time, but anyways, I'm going to stop the simulation in between to show the results. Now our simulation has run, everything has happened properly. There is files generated. But these trajectories, if you directly load, let me show you how do they look like. If you load the trajectories in VMD for analysis, for example, I'll explain how to use the VMD and all when we come to the analysis. But we need to transform these trajectories. And why we need to transform? I'm just giving you an example. So when you open any trajectory, so first we need to load the GRO file. Let's say mdm-ions.gro, the initial configuration we can load. And when we load the trajectory file, let's say NVT trajectory that we use. So if you load, you will see something like this. This is all the lines and everything that you're seeing. So these are the artifacts caused because of the periodic boundary conditions. What it means is when we were looking at the periodic boundary conditions, what happens sometimes, let's say there is a water molecule here. So hydrogen, oxygen stay in the same box, but one of the hydrogen goes in the another box. So what happens when we load the trajectory? Only the central part is moved. Now this atom of this box has already moved over here. From here it has moved out. From here the same atom has come into this, and the same atom from this box has moved into this box. So what it does, it connects these two atoms and forms a line because it thinks this atom belongs to this molecule. The numbers are given that way. So that is the artifact because of the periodic boundary. So we need to transform the trajectories and make sure that we are wrapping this back. So what the wrapping will do, it will place all the atoms in their respective original position. So this atom will go over here, the moved atom will come here, and the original atom of the hydrogen will be attached to the hydrogen. Even though it is outside the box, it will still be attached to it. So this way, the periodic boundary conditions can be handled. If you don't handle them, this is the kind of things you will see, which is not correct way of visualizing your trajectories. So let's transform the trajectories and see how to do that. So for that, the Gromacs, now our analysis is going to start. So for this, the Gromacs has provided inbuilt command called as a trajectory converter, or TRJ CONV, that can be used to convert the trajectories. So I'll show you how to convert these trajectories so that our periodic boundaries can be corrected, and then you will be able to visualize these trajectories in a much better fashion. So to transform it, again the commands will be same. So we're going to start from the very first. Energy minimization is okay. Even if you don't use that, it's fine. But the all MD run we will check. So the command is like this: GMX TRJCONV, that is trajectory converter. The first, it needs the TPR file that was used, so it can get all the input. So for earlier run, it was nvt.tpr, the first MD run. Then it requires the current trajectory. So our trajectory is nvt.xtc. Then what should be the name of the output trajectory? Let's call it nvt corrected .xtc. So it will reform these coordinates and make it proper. Now, how do you want to convert? What exactly you want to convert? Do you want to convert it into a different file type? Do you want to convert it into PDB, and what? We want to just take care of the periodic boundary conditions. So only thing we have to give is the PBC, that is the dash or hyphen PBC. So it will automatically correct the periodic boundary conditions. And around what you want to wrap the atoms, you can say mol, or there are many other terms. You can wrap it around just the protein, you can wrap it just around the ligand and so. So when you say mol, it is going to wrap around the entire solute. Only the solvents will be corrected wherever they are. Next, you also have options to kind of make the trajectory a little bit compact. So for that, you have an option called UR, so that your size will be a little bit reduced. Unnecessary data will not be included. So this way you can reduce the data, and you can just simply enter. It will ask out of all the groups, which groups you want to output into the final trajectory. For now, we are going to use all the system, even the water molecules. Usually what people do, they only use the protein and ligand, and the rest of the molecules they will not use for the writing the new coordinates. But we are going to use the entire system. So let's put 0 and enter. So the 100 frames or 100 picoseconds have been written. Next, do this exactly same for the NPT as well. So npt.tpr will be input. The output will be the npt.xtc. The corrected output file will be the npt.corrected, and the PBC will be the same. And we will also compress the trajectory. Enter. Select the system. So 200 frames of this will also be given. Now our this simulation is gonna take some time to run, so I'm gonna just stop it in between, and whatever has been finished, I'm gonna use it. So it has finished running around 253 steps plus something. So we will just use those many just to analyze, so that we can. So I've stopped the simulation. You can see the performance and all. Now the same thing we did for other trajectories, we also have to do for the simulation trajectory, so that we can have the proper results. So our simulation trajectory is named as MD ligand or MD underscore lig. So use the same nomenclature over here for the input files, for the XTC files, and for the output file. This will be MD underscore corrected. Okay, the same system. So there are only 50 frames so far because we were saving it every 500 picosecond, every 10 picoseconds, not one. So we only have 50. So that's fine. We can just use it for basic analysis. So now our entire simulation has run. We have the data. What to do? What next to do? First thing, we can do the visualization. So now we are going to learn about the software called VMD, which is used for the visualization of the trajectories. So you can see here VMD, when you open, it opens as a multi-component window, which contains the command line, the main control window, and the visualizer window, where you will see, interact with the molecules. It's a very powerful software, and it can handle the trajectories from many commonly used softwares for like Gromacs, Amber, or Desmond, NAMD, Charm. All of these files it can support for analysis. Along with visualization, it also provides a large number of analysis toolkits to perform. First, let's simply visualize the trajectory and see how or what changes have happened to our system over time, how molecules are moving. So first, we have to open by using the file new molecule, the main configuration file, which we use as an input. Our initial system was called as mdmions.gro. So this will be used. This is what our initial system is. Let's try to put it in a very simplistic representation by using this option of graphics representation. Here we can select for every type, every part of the system, what type of representation to use so that we can make some changes. I'm only going to put protein and ligand for now. Water I won't keep here right now for visualization. So that, and this will be, let's call as van der Waals. So this will be the end of the simulation trajectory, and a basic visualization will be kind of, looking at it. Let's close this box. Now, whenever you are adding the trajectory, it has to be added to something. So you have to make sure that you are adding the trajectory to already loaded molecule and not a new molecule. If you load it new molecule, it will not load anything into the system. So let's first use the corrected NVT trajectory, that is the heating phase. So NVT corrected .xtc and load it. You will see that there are some small movements. The atoms are getting movement and velocity because of the heating. Next, to the same system, let's load the NPT corrected trajectory. All right. Oh, I think I already loaded the NPT. I should have first loaded NVT. Anyways, this is wrong actually. Do it in the opposite way. So now the next will be the final MD run. So far you can see the protein and ligand are not moving much, they are restrained. But in the final MD, they were not restrained. So they will have the complete freedom to move, and you can see they have started now moving a lot. Now this is a very small simulation, so it's just for example. So we have loaded. Now we can, once loaded all the frames, you can now slowly observe how the ligands are moving, what changes are happening. You can also track different bonds. So for example, let's try to create a new representation. Let's keep this as the maybe licorice, yeah, and then create a new representation which will okay, and then you can run your trajectory. So you can see how the trajectory is moving. If there are any hydrogen bonds formed between, you will be able to visualize those as well, and many things can be seen. So this is where you can see how your atoms are moving, how stable it is, over what time it is staying stable, after how much time it can go out of the system. So this is basic visual analysis. Now we will go to the numerical analysis, like calculating the RMSD of the system, then calculating the root mean square fluctuations, and many things that can be calculated. So for calculating these parameters, which are the key for the final MD analysis, we can either use the Gromacs toolkits, which are there. A lot of things are there. or you can also use this VMD, in which all these options are also available. I'll show you both the ways of getting your calculations done, because visually it just shows the movements of the atom, but exactly what is happening with the system, that we can use by different mathematical terminologies like RMSD, or that is root mean square deviations, or you can use things like RMSF, root mean square fluctuations, and so on. We can also add one more representation here for water, because water is going to be extremely important in this entire thing. So we'll see how the hydrogen bonding between water and other molecules happen, and so on. So with water, again, you can have different bonds formed. And then you can run it again. So you can see the changing dynamics of the water, the hydrogen bonds between water and other molecules, and the ligands, how they are formed, how they are broken. You can also select a specific region for tracking the hydrogen bonds, which also will be doing just in a little bit. So let's pause for the VMD a little bit and go back to the Gromacs. All right. So the next thing we need, that the RMS plot, that is the RMSD. So how do we calculate the RMSD between the entire? So what is first of all RMSD? It's a root mean square deviation, which tells us how much the system is changing over the time of the simulation. That's what it shows us. Okay. So now when it comes to using GMX for RMSD calculation, the command is very simple. It's called as RMS. It's a part of the Gromacs that you can use for generating the RMS graphs. It requires two inputs. First, your TPR structure. So for us, it's ligand.tpr. The second, it needs the entire trajectory. So for entire trajectory, it is going to have it. Our is, let's see the corrected trajectory. Only with 50 frames, we'll see what happens. And then you can have the output file. Let's call it rmsd.xvg. So it will ask you for which group we should use as a standard or a reference. Let's keep it. We are only want the protein RMSD. So let's keep it protein. Then what you want to track with respect to protein over the rest of the simulation, you can use protein or ligand or protein ligand both. For now, let's check with the protein. Now this RMSD graph you can always plot by using the XMGRES, just the way we have been doing for all other plots, and you can see how the RMSD is changing. So we only have very few frames, only 50 frames, so we don't have like much smooth graph. But if you save the frames like every picosecond, we will have a much smoother graph over here. So you can see over the time the RMSD is changing. That means there are the conformational changes happening in the protein with respect to the earlier configuration. So earlier there were not much changes, but over the time slowly the protein is changing so much in its structure that you are seeing the more and more RMS. This RMS is not very big; it's just 0.2. Actually, no, it's big. Actually, 0.2 nanometer is still huge. So you can plot all of these things like that. So we can plot the root mean square fluctuations for every residue. You can plot many other parameters that you can require. So Gromacs is one thing. Let's come back to our this part, that is our VMD calculations, and see how do we use this for basic analysis. We are only going to focus on the basic analysis, not anything complicated right now, because we don't have time. We're running out of time. We just have a few minutes left. All right. So now we have the system represented, and we want to find some properties of this. Even water, actually, we can hide for now. We will see water also, but let's not show it in the simulation. Okay. So when you load your system and the entire trajectory into the VMD, it provides you a lot of options. So if you go to the extensions and analysis tab, it provides a lot of options. Of course, all of them may not be applicable to your system, because some of them are for some specific software like NAMD and all, but you can check. The first, like if you are looking for drug ligand interactions, the most important thing you have to check is the hydrogen bond formation, whether the molecule is able to form enough hydrogen bonds and whether they are stable over the period of simulation or not. So let's click on the hydrogen bonds. It will open the hydrogen bond window. It will ask you which hydrogen bonds you want to track between which two sets. So you can have any two atom selections, like for example, protein and water, or protein and ligand, or water and water, water and ligand. So all of this you can track. So we're gonna track all. First, let's see most importantly between the protein and ligand. So how do we select the ligand? Well, there are various ways to select it. For now, I'm using directly the name of the ligand called K23, and I'm going to use it. After this, you can simply click on the find H1, and you can see that over the time of simulation, the maximum hydrogen bonds is one only that is making, but that one bond is more or less stable over the entire range of the simulation. In all the frames, the bond is almost there. Of course, in some during the movements, it's gonna go a little bit away. Atoms are gonna move, so bond will be missed. But over the time, at least one bond is maintained, so that at least that much of confidence you can have in the binding of your molecule. Then let's see between protein and water, what are the dynamics of the hydrogen bonds, and check it. This is going to be, of course, a huge number of bonds. So you can see there are many hydrogen bonds between protein and water and how they are changing. It also tells you about how protein is interacting with water, how ligand is interacting with water, and how both of them are interacting with each other. So we can also plot solvent accessible surface area and see whether the molecule is getting outside into the solvent or still keeping to the binding side and all. So a lot of things we can calculate. Let's calculate between the residue, that is ligand, and water, and check whether there are any hydrogen bonds. Sometimes ligand can also have the hydrogen mediated interactions with proteins. So you can see here between the ligand and the water also there are one hydrogen bond at least. So that one hydrogen molecule, the one water molecule in the vicinity will be very important for its binding to the protein. So that can also be seen. So all of these things you can do with basic analysis. You can also plot your RMSDs and calculate by using this tool. There is a symmetry tool, the RMSD tool you can use. So RMSD trajectory or RMSD calculator, in which you can have select the residues for which you want to generate the trajectory. First, you can also align it to the first frame and then calculate the RMSD. Actually, no, this. So this can give the same thing, what we already have seen in the RMSD graph of Gromacs. The same thing can be there. You can also generate RMSD of over the trajectory of many different like types. So you can have trajectory before alignment, after alignment, and so many other things. So we can also select protein, let's say, or rest name K23. So the entire protein ligand system we can have. So let's first align and then calculate the trajectory. So it will have the average RMSD over the trajectory is 0.773, which is not very huge. That means the overall the complex is stable, at least for this simulation. You can also plot the RMSD. So you can see initial sometime the protein and ligand are quite like stable, but at the end suddenly there is a sudden jump into the increase in the RMSD, where something is changing. Maybe the molecules the moving out of the protein or going a little bit away from the protein. That could be possible. Let's check only for the protein the same thing. How do we look at the graph? Sorry, a line. Yeah. So same thing, you can see the protein is changing some conformation, some significant changes are happening. Again, we can have those by using the average structures or by doing the PC or principal component analysis or directional reduction. So these are the things can be done by using this. So now I think we are almost out of time for now. So we are just gonna have a quick wrap up of this entire thing. So if you check the entire GMX profile of all the types of commands which are available, you will see to do a lot of different things. You have commands like, for example, you have commands for analysis of solvent accessible surface area, which can be a good indicator of your dissolution potential of the ligand and how it is. RMSF can be a very good indicator of how stable each one of the residue is. You can also track the binding set residues and see how flexible they are and which molecules may be giving the instability, and all those things you can see. You also have things for the PCA calculation called principal. So you can calculate the principal axis So what it will basically cluster your the entire trajectory, and only the important type of conformations, the minimum energy conformations, will be taken forward for that. And many others. You have things like clustering, like G cluster or cluster. We can generate the radius of gyration, and many calculations can be done from within the Gromacs. You don't even have to go for any other thing. But again, those who prefer the graphical interfaces, the VMD can be given. So that's the type of introduction I wanted to give you, how to run and set up your system. Now, coming to some of the questions that have been posted earlier, let me go through one by one all of this, and also discuss importantly, the configurations and all. So one of the question was, share the instructions how to download VMD and command files, as well as how to install. It will help us. Yes, I'm going to share the entire setup or the example files and everything. You can find it after maybe 1 hour in the description of this. I will share the link in the description of the same video. Just come to the same link on YouTube, and at the description you will find all the data, the commands list, installation instructions, everything. How to do implicit solvent. So for implicit solvent, if you do the GMX command, the PDB to GMX, if you remember, PDB to GMX. So in this, there is one specific tag called as, where it is, for implicit solvent. Okay, actually water you can say none. At this point, you can say water to be none. So if you select water none, it will go for the implicit calculation of the solvents, the mathematical expressions. You don't have to use any water molecules for the solvation. Okay. Now the next question is, apart from water, how to take other solvents? You can take the same way. So only thing you need the actual structure coordinates of one type of atom, and you can use, like, there are command line tools like PackMol, for example, which will be able to fill the entire box with that. So solve the when you have that CS tab, I'll go back to that command. Whenever we have used the solvation, the solvate command. So in that solvate command, you can see here we have the configuration of solvent. So here it will be the Gromacs or GRO file or coordinate file of that solvent. Whatever you are using, the file will be that. So that will be filled in the entire simulation, entire box. Mixed solvent models. Mixed solvent models can also be done in a similar way. Just like for every type of solvent, just specify this and randomly it will be filled. But for mixed solvent, it is recommended that you first use a PackMol or other type of softwares to pack the entire unit cell that you are forming with the mixed solvents the way you want, the distribution, the density, and then use it. Not use this directly for solvation. Kindly, can you again explain the graph interpretation of RMSD and RMS, please? Yes, I'll come to that. And also tell us how to calculate the average values. Okay, so first of all, going back to the RMSD or root mean square deviations, they tell us about how the structure is changing from a reference structure. Now, usually the reference structure is taken as the first frame, the 0th frame, and how it has been changing. So distance between every atom is calculated, and the root, the mean and the root of that is taken. The squared root basically will be taken, and that will give us the RMSD value. So more the RMSD value means that the structure is changing a lot. The atoms are moving too much from their original positions. Low RMSD means the structure is very intact and very stable, and there are not a lot of movements of the atoms. That's what you see. Now, there are not standard values as such. Every system, it could be a different value as a cutoff depending on the size and the system configuration. So that way you can read. How to calculate the average values? Average values will be calculated again by many ways. So within the Gromacs toolkit, you have GMX help commands. So there is a command for average calculation, average structure calculation. Either you can use cluster or you can directly use where is that average? Okay. Or in VMD, you can do that actually. VMD will be much easier to use for this purpose. You can go to extension. Okay, my VMD is hanged. Okay, so I am not sure in JMX which command, but yes, there is one command which can generate the average structure directly from the given data. But in your VMD, you will find that option in the extensions analysis directly to generate the average structures. Average structures can also be generated through the clustering. So cluster, what it will do, rather than giving you one single average structure, the G cluster, this cluster command will give you the small configuration sets, and in each one of them, you can find one representative structure. So you can explore the average structure in different minima, the local minima, the global minima, all sorts of minima you can explore. So that can also be good way to getting the average structure. Yeah. So now let's come to the one important question that many of you have asked about the configuration. So configuration of the system that you need, it depends on the size of the system you are going to simulate. But if you are going to use a laptop or a desktop for your thing, for the simulations, it is recommended that the RAM, you should have at least 16 GB of RAM to at least handle big complexes. Then the processor should be good enough, at least a new generation i7 or i9 processor if it is a desktop. The cores of the processor matter. If you use a octa-core processor, you essentially get 16 threads to work on at a time, or 16 cores. So it will be much better boost. But most importantly, for MD, many calculations are done on the graphical interfaces, the graphical processor units or GPUs, essentially the graphics cards that we use to play the game. So the most recent and powerful graphics card is important. If you're using a laptop, you can go for the graphics card starting from the 1660 series, 1660 Ti, or the 2020 or 2021 Ti series, which are very powerful and multi-core, so that can speed up your simulations quite significantly. Apart from that, it is also expected that your space is good, at least 2 TBs or so, because every simulation can generate enough GBs of data. So your space might get quickly spaced out. And also important is that your hard disk that you're using should be of good speed, reading and writing speed. Otherwise, it will be very troublesome to copy and paste files, edit, read files. It takes a lot of time to do this. So for getting this performance, my laptop is not a very optimal for MD simulation, but what I'm currently using is a 10th generation i7 processor, which has 8 cores. Basically 16 threads can be done. Every core is a dual core. So we get a 16 threads, the 16 GB of RAM, and the graphics processor, which is 1650 titanium or Ti series. So that gives me 126 nanosecond per day of performance for this protein, which contains around, I don't know, 27 or so amino acids. But quickly, when you increase the amino acid, this will change drastically. So yeah, recommended hardwares are very high performance clusters of GPUs like Titan series or Quadro series or GeForce higher series and so on. So for desktop, you can build a very good system with all those things. Yeah. So with this, let's come to the end and wrap this thing. I hope you are at least been able to understand the process. I know that in one go, you may not be able to run it on your own directly, but with some sample files and example files, you can start running it on your own with example data, and then you can try to extend it on your own systems. If you do not have access to the high performance compute for bigger systems, every country, every government always has at least some universities in your region definitely may have these kind of compute clusters free for academic usage. Try to search them and approach them for the access. They will provide you with the access to their servers, and you can upload your data there for running. Linux is the most preferred hardware, the operating system for the running Gromacs or others. If you use the Gromacs on Windows, it works very well and very smoothly, but it cannot use the GPU. So even if you have a graphics processing unit on Windows, it is not able to use it. So it will not be optimized. So it's always recommended to install the Linux or Ubuntu or similar systems to run the Gromacs simulations. Same for Desmond. Desmond works only on the Gromacs. Desmond works only on the Linux system. So you will need some kind of a Linux system to run the Desmond GPU version of it. So this is the kind of recommended architecture you should have for running basic simulations, and that will do plenty of work for you. All your single simulations can be done. but it is your daily job and you want like lots and lots of simulation, it's not good to put your computer or laptop through such a huge harsh computational conditions. So better go for a desktop or a cluster. All right. So thank you everyone, and I say goodbye here. We can take another 2 to 3 minutes for having any questions. You can ask those. If not, then we can end the session right here. Thank you everyone for joining, and stay tuned for more such videos around the MDAs. The free energy calculations will be coming, and a detailed analysis videos will also be coming soon. So just stay tuned, and see you soon. Interact with you soon. All right. Goodbye everyone, and have a safe time wherever you are. Stay safe, stay protected. Question. Okay, so the question is about again showing the graph. Okay, sure. I'll show the RMSD graph one more time and we can see what's happening here. So let's check it. So the graph we had generated for a protein earlier. Let's use the same one, and by XMGRESH, the RMSD plot. So this was only for the protein. So this plot that you're looking essentially shows that what is the change in the protein structure from the first frame, the first coordinates initial. So of course, for the very first, it will be 0 because if you subtract the coordinates of the same system from itself, it will be 0. So it will start from 0. The next frame, whatever movements have happened, how much are those movements and how much the structure has changed from earlier, that will have. So as you can see in the initial pages of your it is actually the first 100 frames are of the NPT and NVT. So it is changing the structure a little bit. After it is equilibrated properly, after around 300 or so, after that you can see there is not much change in the protein structure. So overall the protein structure is same, but earlier during the equilibration, it has definitely, actually it's not equilibration even, but it has changed only a little bit. But after that, it has been stable for rest of the simulation. Initial only there has been some changes. So if you finally calculate the RMSD value, if you run that RMSD command one more time, just to see the example, which was the energy. So GMX energy, then we have the file of EDR file and the output. Actually, RMS only will use. Corrected dot trajectory and minus 2 RMSD. So it will ask you for the reference system. You can use your system or just a protein, and then you can plot the protein against it. So we'll see the total RMSD calculated. You can also check the RMSD just of the main chain of the protein. So you can also see the side chains, rotations, and so on. So all of this can be estimated by using this plot. So the same plot for ligand could be different. So now let's plot the RMSD for a ligand. How much ligand is changing from its original position. So this time, let's create a file RMSD ligand. It will ask which group. Let's take 19. That is our ligand, and 19. So this plot will look something different than the protein, definitely. XMGresh, RMSD ligand, and let's plot it. So here you can see the plot for RMSD of the ligand. Again, you can see there are not much changes. The maximum change is around 0.08. So the ligand has not changed substantially over the time. So this is why you can interpret. Can we use virtual environments? In my case, I have Windows. For Gromacs, you don't need even virtual environments. It will be difficult to connect to the graphic unit. So rather, you go ahead and do the direct installation. As I told you, within next 15 minutes or so, I'll be uploading all the installation instructions for Windows and all for Gromacs. So it's very easy to use on Windows. But whether you use VM, it will be much more complicated. Or whether you use the Linux subsystem. In both ways, on Windows, it may not be able to fully utilize your hardware, the graphics units. So that's the only issue. Otherwise, that's all will be fine. Okay. So thank you so much, everyone. If you have any more questions, you can keep commenting on the video. As soon as possible, I'll give you answers to those. Some questions which I have missed earlier, I'll go through them later, and I'll make sure that you get the answers to those as well. And we'll soon come up with more videos on the analytical part of it, because we are not able to complete a lot of analysis today, but we'll definitely discuss that soon. And goodbye, everyone. I'm ending the session now.